Welcome to the Global Health, Biodiversity, and Therapeutics Hybrid Seminar organized by Harvard University Herbaria and University of Costa Rica. Thank you for joining us today. I am Jimena Morales, professor and researcher at the School of Pharmacy and the Pharmaceutical Research Institute, and today I will be hosting this seminar. As you may know, this seminar seeks to address the potential for the development of new therapeutic products from the American continent's biota, as well as to promote collaboration between research institutes in the region to generate advances in these areas. We will explore innovative ideas and share experiences in the next themes: the search of new natural products for diseases, intellectual property, funding opportunities, extract banks and local capacities, and natural product supplements. We hope you enjoyed this seminar very much. Without further ado, we will begin with the opening remarks by Master of Science María Laura Arias Echandia, Vice Rector of Research at the University of Costa Rica.
Morning to all of you. Welcome to University of Costa Rica and welcome to Universidad de Costa Rica. Okay. Acercarme. Okay. Dr. Luis Esteban Hernández, Pharmacy Faculty Dean, PhD Donald Pfister, ex-director of the Harvard Herbary and curator of the Herbary and Fairless Library, Art and Science Faculty from Harvard. Welcome. PhD Ryan Nett, Assistant Professor from the Art and Science Faculty from Harvard. PhD Veronica Vargas, Associate Researcher from the Art and Science Faculty from Harvard. Dr. Germán Madrigal Redondo, the Director of the Pharmaceutical Research Institute. Dr. Giselle Tamayo Castillo, Director of the Research Center on Natural Products from Universidad de Costa Rica. Dr. José Alberto Gatkins, President of the Pharmaceutical College in Costa Rica, and to all persons that are here and also by the web. For thousands of years, nature has been a source of medical substances, and lots of contemporary remedies have been identified from natural origins. Plants have long been used as for herbal medicines to treat various disorders, and the different natural products have inspired the design, discovery, and development of new drugs. Scientists' investigation of medicinal plants has been crucial in uncovering early drugs, each possessing distinct pharmacological characteristics. Few examples of the use of plants in medicines include paclitaxel, obtained from Taxus bifolia, used in the treatment of lung, ovarian, and breast cancer. Artemisinin, derived from the traditional Chinese herb Artemisia annua, used to battle resistant malaria. Digitoxin, extract from the foxglove plant, used for congestive heart failure. Cocaine, first known for its anesthetic effects, was traditionally used for local anesthesia and constriction of blood vessels. Codeine, an opioid alkaloid derived from the opium poppy, gained significant popularity due to its pain-relieving and cough-suppressing qualities. Quinine, derived from the bark of the cinchona tree, played a crucial role in the fight against malaria. And notably, several early drugs, including digitoxin and quinine, are still employed in contemporary medicine, illustrating the continuing significance of plant-derived substances. Many plants have been used as remedies, as medicines, still can be used, and many other ones can be discovered in the future years for the treatment of the very different diseases, including cancer, including mental diseases, stomach diseases, etc. With the invention of recent molecular targets based on proteins, there is a growing need for fresh chemical diversification in screening. Natural products will play a vital part in supplying this need via the continuous exploration of global biodiversity, the majority of which remains unexplored. Costa Rica is a very small country. It accounts for only 0.03% of the Earth's surface. We have around 51,000 square kilometers. However, it contains nearly 6% of the world's biodiversity. It's considered as one of the 20 countries with the highest biodiversity in the world. Chemical diversity and biodiversity are the two sides of the same coin, where plant products offer a vast repertoire of chemical diversity, which in turn may provide an array of lead structures. Interestingly, most of the therapeutically active molecules are plant secondary metabolites, capable of interacting with a diverse range of macromolecules such as proteins, DNA, and thus exhibiting important biological functions that can be used to yield biomoleculars of therapeutic importance. This seminar is focused on the potential development of different natural compounds that can be used for the treatment of diseases including cancer, dementia, and non-attended diseases. The research centers participating, INIFAR and CIPRONA, are research centers that have as one of their main objectives the study of different natural products that can be used for the formulation of new medical products that can be the answer to the devastating chronic diseases mentioned before and others. Since 2023, INIFAR has established a strong research collaboration with the Herbary from Harvard University, and this has allowed the development of this global health and therapeutic seminar with two main objectives that include the potential development of new therapeutic products, as well as the promotion of collaborative activities between different Latin American research institutes focused on biodiversity and therapeutics. I want to give all of you a warm welcome. I am sure that many interesting research proposals and networks will be derived from this meeting. As part of UCR, I can say that our researchers are very good, devoted to their work, and eager to find new solutions to all problems. So I am sure that successful conclusions will be obtained from this seminar. Welcome to your university, your house, and to Costa Rica, a small country in size, but rich in biodiversity and innovation skills, direct to the main objective of having a better world. Welcome. I hope you have a very nice stay. You learn a lot. You do a lot of research networks, and if I can help you in anything, I will be very glad to participate. Welcome, really. Thank you very much.
Thank you for your words. Dr. Luis Esteban Hernández, the Dean of the School of Pharmacy of the University of Costa Rica, will now proceed to give us some words as well.
Thank you, Dr. Morales. Good morning, PhD Donald P. Pfister, ex-director of the Harvard Herbary and curator of Herbary Follow Library. Dr. Ryan Nett, assistant professor, art and science of faculty and pharmacy, Harvard. PhD Veronica Vargas, associated research, art and science faculty, the University of Harvard. Dr. Germán Madrigal Redondo, director of Pharmaceutical Research Institute. Dr. Giselle Tamaño Castillo, director of Research Center of Natural Products of the University of Costa Rica. And Dr. Jose Alberto Gatjens, president of Pharmaceutical College of Costa Rica. Today, we begin a series of presentations related to the topic to phytotherapy. The use of natural products to treat different health problems opened the doors a series of investigations where Latin American countries can provide about raw materials and trained professionals for discovery findings and improve people quality of life. Welcome to this activity, and thank you to the researchers who heard these findings. Topics such as cancer prevention with phytochemicals, the use of natural extracts to treat dementia or COVID-19 will be exposed in this activity. I hope that this activity will be to benefit for you, and thank you will be able to create collaborations with your working groups. Good morning and welcome to Costa Rica, and I like that you approve this activity. Well,
I will now invite Veronica Vargas to be the host for the rest of this morning. Welcome.
Good morning to everyone. And now I'm going to introduce you to Professor Don Pfister. He is an emeritus professor, former director of the Harvard Herbarium, and curator of the Farlow Library and the Herbarium at the Harvard Faculty of Arts and Science. His research focuses on biology and systematic of fungi. Welcome, Professor Pfister.
Thank you, and welcome on behalf of the Harvard University Herbaria. We're delighted that the interests and activities of this network have continued to grow and interconnect this community. This is the second seminar in this format, and we are delighted with the participation both in person and online. Last year we met at Harvard. This year we're delighted and want to thank the organizers at the University of Costa Rica for holding this event. We often talk about hotspots in our study of biodiversity, but I look at the topics we are discussing today and in the community as dark spots, that is, topics that are understudied, to explore organisms for compounds that can direct exploration for beneficial health and medicine. That's the goal, and and goes along with the proper identification of the organisms involved. From the law to the field, to the lab, to barcoding for identification, all are important. I'm sure that you're going to hear many, many topics along these lines of great interest to this community. I wish you all an exciting and fruitful day. I wish I were there. I just returned from a conference in Europe, and I'm sorry that I'm missing this. We look forward to the blooming of new ideas and cooperation. Thanks again to the local organizers and to Tracy Barbaro at Harvard, who has become your pen pal. Thank you.
Thank you, Professor Pfister. And now I will give a little introduction to our project. This is the Consortium for Global Health, Biodiversity, and Therapeutic Innovation, a South to South and South to North collaboration. Next. Natural products, as already have been said, have long demonstrated their therapeutic value. Here we have two graphs. The first one being all drugs approved by the FDA over the past 4 decades. Approx 50% of all drugs are synthetic or semi-synthetic, inspired by nature, the dark blue, or are natural, the light blue, both from marine and terrestrial sources. For cancer drug, at the right, is even higher, the proportion. About half are natural inspired and 14% are natural. So despite progress in drug synthesis method, the complexity of many natural products continue to make them challenging to replicate, leading to their continued use in natural forms, the light blue. Latin America and the Caribbean is one of the most biodiverse regions in the world, including plants, fungi, and animals. According to the United Nations Environment Program, around 60% of global terrestrial life, including water and marine species, can be found in Latin America and the Caribbean region. Central and South America hold around 7 biodiverse hotspots, which definition is at least 1,500 species per 10,000 square kilometers. Species not found elsewhere on Earth, and that also are in danger. These are, for example, the Choco Costa Rica region, the tropical Andes, and the Chilean Valdivian forest, for example. There are approx 350,000 known species of plants worldwide, and many of these remain unexplored chemically. The same is true for fungi, of which 2.2 million and 3.8 million estimated 90% remain unknown, holding potential for future discoveries. Plant and fungi-derived drugs are essential for global health. For example, the World Health Organization list of essential medicine includes drugs inspired by nature, such as penicillin, artemether for malaria, vincristine for cancer, etc. Some of those drugs are rooted in traditional Chinese, Ayurvedic, and indigenous knowledge. For example, quinine from the Peruvian cinchona tree and cat's claw from the Amazon rainforest. About 80% of the global population uses herbs in their health care, a primary source or as a complementary source. The World Health Organization advocates for more research into their safety and effectiveness. Our challenge is systematically compiling thousands of papers from journals and university dissertations using a strong method. Biodiversity potential for addressing global health challenge is immense when combined with new technological advancements such as DNA sequencing, molecular techniques, and artificial intelligence. This is data from a paper that I co-authored with Darrow. Over 26 research and development projects in the region, in the Latin America and the Caribbean region, are ongoing on natural products. About half of them are in the preclinical phase, and only around 5 of them are already in the market, coming from Brazil and Cuba. Brazil leads is attributed to its size, vast biodiversity, and substantial investment in R&D. Costa Rica, Colombia, and Chile follow. Our project was born during the pandemic. With the support of the Radcliffe Institute from Harvard, we host 2 virtual seminars. And in the year 2022, we launched a pilot project with the sponsorship of the Harvard Herbaria with Professor Don Pfister, searching for antivirals against COVID-19. In collaboration with the Rega Institute at the University of Leuven in Belgium, the Salk Institute, the Argentinian Ministry of Science, the National Patagonian University San Juan Bosco in Argentina, and INDICASAT in Panama. Our partners from California and Argentina prepared 200 extracts from plant, algae, and fungi, which were tested for antiviral activity against COVID-19. We got 4 hits, marking an aha moment of our project. We engage in scientific diplomacy, supporting collaboration among scientists, scholars, labs, policymakers, practitioners, and entrepreneurs. Our goal is to translate scientific discovery into therapeutics that reach people promptly and equitable. We support R&D in 3 interconnected areas: basic research, applied research targeting specific diseases, and upgrades in traditional medicine. This morning, we will have several presentations on basic research, on applied R&D targeting specific diseases such as neglected tropical diseases, cancer, and dementia. Other presentations will touch on research accelerating tools such as a large bank of extracts, improving ID through sequencing, and using computational tools. Latin American and the Caribbean universities are leading the way in researching natural products for medicinal properties. Their work involves R&D on native medicinal plants, psychedelics, and local marine organisms. After lunch, we will have several presentations from LAC researchers. In the past, we collaborated with the Rega Institute from the University of Leuven in Belgium. Soon, we plan to collaborate with the Fundación MEDINA and the US National Cancer Institute. Over-harvesting wild trees, plants, and fungi for medicinal purpose can lead to species decline and even extinction, especially in fragile biodiverse hotspots. One example is paclitaxel, who was already mentioned this morning, a drug widely used for breast cancer treatment. It was initially derived from the bark of the Pacific yew tree and is led to harvesting thousands of trees, leading to a 30% decline over time of the population tree. As a result, the Pacific yew tree is now classified as near threatened. To promote sustainability, in vitro cultivation techniques can be employed for the industrial production and commercialization of natural drugs. Today, Botanical Solutions, this company, will serve as an example. Educating and building infrastructure like herbarium, medicinal gardens, and databases naming plant species is important. The Kew Royal Garden scientists have identified dark spots in our biodiversity knowledge, particularly in the Andean regions, Peru, Ecuador. These dark spot regions lack information about their biodiversity. We want to collaborate with the Royal Botanic Gardens Kew to address this knowledge gap and support research and conservation. Finally, I'm going to say something about intellectual property. Terrestrial and marine resources follow distinct intellectual property regimes. While terrestrial biological resources fall under national sovereignty, as agreed in the 92 Convention on Biological Diversity, the legal status of marine resources is less clear-cut. The Convention on Biological Diversity, the Nagoya Protocol, and associated benefit sharing agreements rule the access to terrestrial genetic resources. These resources, plants and animals, cannot be patented as they are product of nature, rather than human invention. Equally, ancestral knowledge, like traditional medicinal plants, cannot be patented due to its roots in community traditions and therefore lack legal novelty. As agreed in the recent diplomatic conference on genetic resources held by the World Intellectual Property Organization. Usually, the intellectual property discussion focus on physical samples, but advancement in genetic sequencing led to the surge in digital biodata. Sharing genetic information across borders has become easier, but ensuring equitable benefit sharing, especially for marine resources, remains a complex challenge. As the digital age transforms bioresearch, reconciling the need for open scientific collaboration with the principle of fair benefit sharing for physical and digital resources is a critical issue for policymakers and researchers alike. I invite you to visit our website for more information about our projects and our collaborator. Thank you very much.
Thank you for all your words. We invite the members of the table to join the rest of the public. And we will go on with all the presentations. We are having a little bit of trouble with Jorge Cabrera, so we will proceed with Olga's presentation. It is titled The MEDINA Foundation Pioneering Work in Natural Products-Based Drug Discovery for Neglected Diseases. This foundation is dedicated to discovering drugs for neglected diseases overlooked by pharmaceutical companies, such as antibiotics and antiparasitic molecules. Welcome.
Hello, can you hear me?
Hi, Olga. We are watching.
Hi, hello. Yes, can you see my slides? I'm already full screen.
Yeah, we are watching.
Okay, thank you, because I don't see you. So thank you so much. It's a pleasure to be here again. Unfortunately, this time I cannot be in person. And thanks so much for the invitation. So I would like to start this presentation just for those who are not familiar about MEDINA and who we are. We are based in Granada, in the south of Spain. And we are a research center focused on drug discovery from microbial natural products. We originated from the former Merck shop and site, had been operating in Spain for 50 years, and MEDINA was launched in 2008 from the assets of the center. And our major focus is the discovery of novel bioactive compounds and new therapies for medical needs, I mean, in different areas. In fact, one of our assets are the large microbial collections that we have. We are working with bacteria and fungi from all over the world. And the libraries that we have been generated have been brought from Merck, but also that in the last 15 years, they have been continued to expand at MEDINA, just to be used in our high-throughput screening platforms. And that's the reason why we are one of the high-capacity center for high-throughput screening and one of the members of this research infrastructure in Europe, European Screen. I will come back to that later on, just to give you some information that probably is of interest to all of you. So in a nutshell, it makes no sense not just to mention that, but I think it's important just to mention the pivotal role of natural products in drug discovery. all of us are convinced here today about the relevance, but also even today, I mean, there's been a time that people were just abandoning the use given the difficulty to work on these molecules, but they are really outstanding scaffolds for inspiring chemists to evolve and expand the diversity of chemistry around these molecules. There is, of course, a new interest in these compounds. And we have underexplored sources of potential novel classes, especially in the microbial world. This is where MEDINA is focusing. So in our case, as I was mentioning at the beginning, we have our culture collection, we have the natural products libraries, but also we are just focusing on expanding the genome information of our strains. We have more than 30,000 bacterial genome that can be used for mining. We are now expanding just the whole genome sequences from the high interest strains. But of course, the major starting point are the strains themselves that we have been collecting from all over the world, all compliant with the Nagoya Protocol, and that we are continuously expanding in terms of exploring the potential that they have to produce novel molecules. So in this sense, as I was mentioning before, our discovery programs are focusing in 4 major areas: infectious diseases, neglected tropical diseases. We are also focusing on cancer and neurodegeneration. But today I will only focus on infectious diseases and neglected tropical diseases as examples of what we are doing at MEDINA. And in this case, of course, we are, as many other centers performing drug discovery, we're combining what is the traditional screening, just leveraging the libraries that we have. We continue to generate new assets, new models within these libraries from unique strains that we are just isolating from the environment. And all of this is being processed through our high-throughput screening platforms, just following this high-throughput the replication to engage later on into the bioasset guide isolation to deliver novel chemistry. But in parallel to this approach, there is more and more initiatives, and this is also something that we are performing ourselves in going to more a bottom-up approach, just using all the omics and especially all the genome information about the strains to mine for new biosynthetic gene clusters, just to identify all these talented strains that can be exploited, more following the traditional way, but also just to try to identify potential new pathways that can be delivering novel chemistry. So I will show you today some of the compounds that we have been obtaining, following different approaches. In most of our cases, we are developing our discovery programs in collaboration with many partners, such as in the case of these interparasitic compounds that we identify through the collaboration to the Parasite Center of Excellence that is also based in Granada. Together with them, we have been establishing a platform that has been just providing the capacities for screening for 3 neglected tropical diseases in Spain. And these are just molecules that have been coming out from the screening of our libraries, such as this Straceria geligonata compound, a strain of fungi, this basidiomycete that was delivered in this Stracidolite family of macrolides, or the Pipeloscolisporin that was just also exhibiting good activity on Trypanosoma cruzi and Plasmodium, or more recently, just this antiparasitic exolipopeptide that also exhibited a good activity against Trypanosoma. All these molecules have been just identified from the different strains in our collections and following more traditional alteration approach, screening large subsets on the assays and then deconvoluting and isolating the pure compounds. More recently, we have been identifying a very interestingly one of the new terpenes coming from this marine basidiomycete that had been isolated from in Singapore. That was in our collection, this Alophysina villosa, that were just cultured in just on a rice-based medium that most of you know that is extremely prolific for fungi to produce secondary metabolites. And in this case, we identify a new family of bisabolane type associated terpenoids that were identified the structure, just discovering that were contained in the very rare alleles that are not so common described in nature. The compounds from this family, from this chemical class, have been described as cytotoxic and with antimicrobial activities, but so far no antiparasitic activities have been reported for compounds having this structure moiety. So most interestingly was one of the analogs obtained from this strain. There is vilozane A, that in fact was one of the most potent and also showing activities in the nanomolar range against Plasmodium, and very, very potent as well against Trypanosoma. So this is one of the latest identification, and especially the chemical characteristics of these compounds that we will be just exploring if some development can be done, given that these compounds did not have very pharmacokinetic good activities and properties. When we are facing the antimicrobial discovery programs and more focused on new antibiotics, of course, we are facing, as anyone in the field, going into how to overcome all the challenges we have with emerging resistance and how to deliver novel chemistry and novel mode of action. So this is the holy grail, just to try to identify this type of compounds. But at the same time, it's also interesting not to forget that in the way to try to identify this type of compounds, it's also very interesting to identify molecules that can be used in combination to extend the lifetime of existing antibiotics in the clinic. And at the same time, we need to have in mind that for any potential development, we need to have low frequencies of resistance. We need to address all these challenges that we have, given the outer membrane barrier, the cell penetration issues that we have, the efflux in the case of gram negatives, and later on in the development, how we are going to be addressing the efficacy in the animal models and all the safety and admit issues. So in our case, we are just targeting, just to identify new antibacterial profiles using both phenotypic screens and target-based in cells. And we are aiming at identifying both broad spectrum antibiotics that can be targeting gram negatives or broad spectrum gram negatives, but at the same time, we are also interested in exploring narrow spectrum antibiotics. At the same time, we have small panels going into gram positives, synergies and potentiators, biofilm inhibition, and we are also exploring how, in which way, using mimetic conditions of the infection niche could be used as well to identify potential novel compounds that we have been overcoming. We didn't see in the standard screening conditions. So this is just an example of one of the programs that we have been running for several years in collaboration with our colleagues from DTU in Denmark. This was a big grant funded by the Novo Foundation. And in fact, we just engaged in a dual approach, just going into the traditional way, just to leverage on a selection of extracts from our libraries, take into account all the knowledge that we have from previous screens, to go through all the programs. But the major focus as well was to try to identify talented strains that could be selected from the collections and dive into the novelty of the potential biosynthetic cluster and identify new compounds that potentially could be developed as new antibiotics. So as a result of this approach, we took as hypothesis that in fact these biosynthetic gene clusters are not widely distributed across all taxa. So we focus on these strains that have large genomes that have higher probability to contain a larger diversity of biosynthetic gene clusters. So we integrated all the knowledge from the genome information generated from the high quality of the genomes of these strains, and running comparative genomics to the existing database or internal databases, targeting specific compound classes. But at the same time, of course, for discovering any compound, you need to produce and express, so that we have have been exploring getting back to more the traditional way, in the sense that culturing strains in many different type of nutritional conditions, just using any kind of effectors. But at the same time, what we don't find that this pathway is expressed, just to try to engineer the strain, the original write-up strain, or just to move to other heterologous expression host. And this, in combination with the metabolomics, that has been the tool to really paint and identify any new molecule that can be expressed by these new strains, had been used to help us in the discovery of novel compounds. So in this case, we used a collection of cyanobacteria to select from the 135 general those that were minor, that had been less explored, but that could be richer in the production of novel biosynthetic gene clusters. And this is one of the approaches that we follow, just to identify novel biosynthetic genes. But at the same time, we explore just minor lineages within the streptomyces that have been underexplored, that could be still delivering novel compounds. By comparative genomics, it helps us just to map the diversity within a given genus. This is just how we were exploring within the Cyptolosporangius species, comparing not only our strains, but also that have been already available in the genome sequence. And then we found out that, in fact, more than 90% of the biosynthetic gene clusters had no homology to those that had been previously described within this species. And at the same time, we were able to map in terms of the type of compounds that potentially this biosynthetic gene cluster were able to produce, what was the diversity according to the different type of genomes. So this information was key to make selections and trying to invest resources in the specific strains to go deeper into just trying to trigger on the expression from the genes. This was done, of course, by using different conditions as nutritional conditions, using resins, trying to trigger just the production by crosstalk between species in culturing, using modifiers. In any way, we were able to generate a large diversity of libraries that were explored against the different type of pathogens. And this is the way we were able to map the diversity of these extracts by using the typical GNPS global network annotation, giving us some idea what the potential networks of potential new compounds could be obtained or were contained in the different extracts. By using these approaches, I would like to give you a couple of examples of the type of compounds we discovered. And this is one case of two families of cyclic non-ribosomal peptides that were encoded by one single pathway. So the first cyclic pentapeptide are on the left, the BE18257, have already been described a long time ago, but of course the biosynthetic gene cluster have not been described. But we were really surprised to see another series of gene encoding, another series of pentapeptides, what we describe as pentaminomycins, A to H. There was a family of 8 pentapeptides differing in the composition of the amino acids that we were focusing on because they were unknown and we didn't know what the potential activity by activity was associated to these molecules. So we purify all of them, and then we confronted them against a panel of gram negatives because we were interested, of course, in this spectrum, and we identify on that only 2 of these cyclic peptides were exhibiting some activity against Acinetobacter. Once the compounds were purified, we were able to assess the MIC of 2 of them that, in fact, they were exhibiting relatively moderate activity against Acinetobacter baumannii, but some structure relationship could be a structure activity relationship could be established according to the differential positions that were located in the cyclic pentapeptide. So this is one of the type of approaches that we have been following. And another example I would like to share with you is just go diving into the genomes of this strain. We ended up with a streptomyces that the biosynthetic gene cluster was very quickly a suspicion for a new acidic lipopeptide for the biosynthesis of this type of compounds. In fact, the NRPS genes and especially the A domain substrate specificity. The analysis suggested that they were incorporating negatively charged residues on one side, and at the same time we were identifying some acyl chain biosynthetic genes, suggesting some fatty acids chain on lipid tail that could be potentially be associated to this strain. At that time, we didn't know what type of molecule was encoded by this biosynthetic gene cluster. So what we did as well is just to confront these genes against all known genome databases. And we end up just finding high homology with vancomycin, which is a calcium-dependent antibiotic. That in fact it was only differing in one of the genes. And in fact, the structure prediction was really based on the high homology with the biosynthetic cluster from vancomycin, and at the same time with the A domain substrate specificity. So instead of having a canonical calcium binding motif that we could be explaining as for other calcium-dependent lipopeptides, we were proposing that in fact this sequence of amino acid could be related with this type of activity and was responsible of the potential calcium binding activity of the compound. So to confirm this hypothesis, so what we did is we isolated the compound. We were able to produce it in a reasonable nice amounts, and we were able to produce three analogs, the dilamarcin, A, B, and C. And in fact, all of them are just share the same motifs. And when we tested with and without calcium against MRSA of Heterococcus faecium, we were able to confirm that in fact the dilamarcin A was exhibiting activity only in the presence of calciums against both pathogens, when we didn't see activity in the absence of calcium. So some behavior that's similar to daptomycin. So these are two examples that are being that I wanted to show you today, just showing that there is different avenues, different approaches to combine the previous knowledge that we may have from the strains in being able to deliver novel chemistry. Of course, we will always be we will need to rely on the unknown potential compounds that can be delivered much more from the classical screening, because of course predictions for biosynthetic gene cluster that cannot be predicted through the canonical ways. So in summary, these are the approaches that we are just following and on our side. And at the end, we are trying to focus more and more on feeding the system and the libraries that we are generating with more talented strain, to getting more information from the strain that we are using to generate the models. And not only just combining the whole genome sequence data, but coupling that with all the information that we can gather from the expression of these pathways, using all the metabolomic tools that we have in-house. And of course, just to have good assays to deliver and just to be able to reveal the potential activities to help us just to guide into the purification of the bioactive compounds. And just to my slide, I would like to mention, as you know, we are part of this high throughput screening network of centers in Europe. But today I would like to focus your attention on two projects that we are developing. One of them is in Berlin. We have a central library, what we call open screen libraries. Of course, there's a large library of commercial compounds that are being used for the different type of screens and campaigns. But today, since the last couple of years, we are focusing on establishing a European academic compound library that is open to all scientists worldwide, to our chemists willing to deposit their compounds. So our target is just to manage together 40,000 compounds coming from full synthesis, but also from isolation. And these compounds, what we are offering is just to profile them on the different platforms that we have to be able to annotate their potential bioactivities. The chemists retain their IP. There is an embargo period. So if you are interested in submitting and getting your compounds exposed to the different type of screens that we are running in our libraries, there is a way just to leverage and get more information about the potential bioactivity and interest in developing these molecules. And the final project I would like to mention, this is also related with an activity performed with European Screen, but not only this infrastructure, but with two other infrastructures in Europe, MBRC and Elixir. So there's 3 of them, coupling a large number of research centers. This consortium is running for 3 years. And this is focused on establishing an infrastructure for marine bioprospecting. This means that establishing all the means and the capacities across the different centers to enable all the screening centers, the small companies, those who have needs in just progressing their programs into the discovery of novel compounds with different type of applications, just to get in touch and to know where who they can address to get the answers for the needs in their biodiscovery and bioprospecting from marine sources. So we will have a lot of networking activities during this project. We will try to engage with research centers and companies in the Americas, in Asia, in Africa. So if you are interested, please just follow us through the social networks and just contact me if you are interested to have some interaction with us. So with that, I would like to acknowledge all my colleagues, the MEDINA team who has been involved in all these discovery programs, our colleagues in Denmark at TTU that have been running for almost 6 years, just these discovery programs on new antibiotics, and at the same time our colleagues in Granada that have been establishing the antiparasitic platform for the different centers and the funding members of our activities. So thank you so much, and I will take any questions that you may have.
Thank you very much, Olga. Here we have already some questions for you. Here come the first.
Hello, Olga, do you hear me?
Yes, I can hear you.
This is Giselle Tamayo.
Hi, Giselle. Yes. Nice to see you. Nice to hear you.
I have one question regarding microorganisms, and it is the following. You're talking about talented strains. So it is best to send you the strains so that you can culture them and obtain the most of the secondary metabolites instead of sending you extracts, because that's an issue, particularly when talking about Costa Rican legislation, because we prefer to always to send extracts instead of microorganisms. So it is the following question: how is actually performing extracts versus strains in your platform in screening?
Well, at the end, just receiving extract is perfectly fine. And the only thing is how you have been selecting your strains. All this information that we are just generating, just mining the genome and trying to identify potential new clusters and trying to map just molecules that can be contained, just to have some selection beforehand without not going blind. Needs to be done in the first step, but later on you need to culture the strain to produce the extract to be tested. So if you can explore your strains from different angles using omics tools, at the end it's just to try to generate for these specific strains the libraries of extract that you need to or you want to test. So for us, receiving extract is perfectly fine. We also have a limited capacity to generate libraries. Of course, this can be done in large scale, but also we are also doing that for our own strain. So if we are receiving extract from other partners, it can go straight into the screens. So this is much faster. But of course, within a collaboration, we are normally exchanging experiences on both sides and trying to see how we can exploit the best as we can what we are seeing. One thing is what you see on the genome, and some other thing is being able to make this translated into molecules. So that's the biggest challenge.
Okay, thank you, Olga. Now we have another question.
Yeah, hi, I'm Tim Thompson from INDICASAT in Panama. Very impressive work. My question is related to how you approach modes of action in molecular targets. How do you identify targets, especially for parasitic diseases or cancer and other target diseases or cell types that you haven't just mentioned briefly? How do you approach the identification of modes of action and molecular targets?
Yeah, well, on one side, there are certain screens that we are including the target in the screen, although this is a wholesale assay. We already know what we are just screening. So this is helping us a lot in going straight for what we want. In the other cases, we are using phenotypic screenings, and this is being done for cancer, but also for neurodegeneration. We are using not only 2D, but also 3D models to be able to be more reliable in what we can then later on translate. But of course, from this stage that we are seeing some phenotype, then later on, we need to follow the most classical approach in trying to deconvolute for the mode of action, just to have just the standard, checking the standard checkpoints and trying to explore the potential just key aspects of this molecule, how this been targeting or hitting the strain. So this is the most limiting part in this sense, in trying to address mode of action, starting from the phenotypic screens. When we can manage to get the target within the wholesale assay, this is really straightforward to go into mode of action, of course. Mm-hmm.
Okay, thank you. Here you have a third question.
Hi, Olga. Good to see you. This is
Hello.
Marcelino. I have one question. Which strategies are you using to activate those cryptic genes clusters that are present in the bacteria? You know, to make them to produce more interesting chemistry.
Yeah, there is no single formula to do that, and this is very much relying on each strain. So we try to combine a wide array of nutritional conditions just to know how the strain is behaving. We leverage on the previous information that we have from the old screens from the different genera. So that's the way we try to expand that. We try to balance without going into engineering the strains when we don't manage to get these expressions. So beforehand is using with these osmotic approaches. We are using as well different type of effectors that we see in the past that have been helpful in trying to trigger certain compounds. In other cases, we are also challenging strains with extracts coming from pathogens of other strains, not going just only into co-cultivation, which is very difficult to reproduce, but also in having signaling coming from other strains that you can most control to be added to these fermentations. But at the end, it's a trial and error approach. So that's you need to be very much interested in this novelty of the compound, just to try to define the number of conditions you will be giving to the strain. But normally, of course, we use many more conditions that with the standard generation of models of our libraries.
Okay. Well, I think, Olga, we want to thank you for your time for your great presentation. And of course, we keep in touch probably we can collaborate in some projects in the near future. Thank you, Olga.
Thank you so much. A pleasure being with you.
Now, the next presentation, Dr. Omar Kucuk from Emory University. The title of his presentation is Phytochemicals in Cancer Prevention. Dr. Kucuk is an oncologist affiliated with Emory University Hospital. With over 20 years of experience, he had made significant contribution to cancer prevention. His primary research focuses on nutrition and cancer therapy, exploring the effects of micronutrients and phytochemicals or biomarkers related to various cancers. Welcome, Dr. Kucuk.
Hello. Can you hear me?
Yes, we can hear you. Yes.
Okay. Thank you very much for this kind invitation to participate in this meeting.
We're not seeing your presentation.
You're not seeing the presentation? I shared my slides.
No, I believe you're sharing something else, but not the slides. Maybe, right?
Let's see. I thought I shared them.
There it is.
Can you see it now?
Yeah, we're watching it. Thank you.
You can see the slides?
Yes.
Okay,
Yes.
good.
Thank you.
All right. So, yeah, I'm a medical oncologist. My interest is in cancer chemo prevention with natural compounds, with botanicals, particularly with carotenoids and soy isoflavones. So I will be giving two examples during this talk, talking about lycopene in prostate cancer prevention and soy isoflavones in prostate cancer prevention. I have, these are my conflict of interest. Prostate cancer is a very common cancer, as everybody knows, but there are very big geographic differences in the incidence and mortality of prostate cancer. In certain areas of the world, like North America and Northern Europe, the incidence is very high, but in Asia and the Mediterranean region, the risk is low. So epidemiologists have looked at certain factors that might be responsible for the differences in prostate cancer risk. For example, in the Mediterranean region, tomato consumption is obviously very high, and in East Asia, soy products are very high. In India, curcumin intake is high. Maybe certain nutritional and botanical agents may have effects on prostate cancer risk. And based on that hypothesis, a lot of studies have been conducted. It has been known for a long time that diet plays an important role in cancer risk and outcomes. A third of all cancers are thought to be due to dietary factors, especially vegetables in the diet are generally associated with a decreased risk of many cancers that are listed at this table. One of my main interests is carotenoids. Giovanici and colleagues from Harvard University, they have looked at also tomato intake and lycopene intake and cancer risk. When they looked at prostate cancer risk, they found that intake of lycopene was inversely associated with prostate cancer risk. But they also looked at other cancers. They published that in 1999, an update of their epidemiologic results. They looked at all the studies that were reported at that time, and they found that there was statistically significant decreases in many cancers. And none of the studies that were statistically significant showed an increased risk. They all showed, in general, a decreased risk of cancer with lycopene intake. What is lycopene? It's a carotenoid found in tomatoes, as well as some other fruits. It's associated with decreased risk of certain cancers. At the lower end, you see the molecular structure of lycopene, which is very symmetrical carbon-hydrogen type of molecule, is lipid soluble. Now, not all tomato products are same. For example, in Giovanici's study, they looked at the intake of tomato fruits and pizza and tomato sauce, which were associated with decreased risk, but tomato juice was not, maybe because lycopene is lipid soluble. Again, not all carotenoids are same. When there's no association with beta carotene, intake, lycopene intake is associated with lower risk of prostate cancer. So in the tomato plant, there are many carotenoids in addition to lycopene, because of the synthesis of lycopene goes through many steps. And these are some of the carotenoids: phytoene, phytofluene, beta carotene, lycopene. These are all found in the tomato. So consuming tomato products may be better than consuming pure lycopene, because there may be some benefits of other carotenoids found in the tomatoes. And tomatoes also have other substances other than carotenoids. Now, Sharoni and Levy from Beersheba in Israel, they did some studies looking at prostate cancer and mammary cancer and looked at certain carotenoids. In this slide, we see lycopene and astaxanthin, two of the carotenoids they looked at. They found that, lycopene is much more effective compared to astaxanthin. And when they looked at patients with colon cancer, they prior to surgery, they gave them lycopene supplementation, and they were able to increase the plasma lycopene. On the right hand, you see the lycopene group and the placebo group, that there is an increase in the lycopene in the intervention group. And also IGF-1 levels went down when the lycopene was consumed as a supplement. That is one of the potential mechanisms how lycopene may decrease the risk of cancer. In this slide, we see some prostate cancer cells that are exposed to testosterone and IGF-1. They increase cell growth, but when astaxanthin and lycopene is present, then this testosterone and IGF-1 induced proliferation is inhibited. In this slide, we again see from Sharoni and Levy looking at PSA levels. Obviously, in prostate cancers, PSA levels is a good marker. As you can see, testosterone increases the PSA levels in these cell lines in prostate cancer cells. When astaxanthin is added, it decreases the PSA production, but lycopene is much more effective. And combination of lycopene and fluorine, phytoin, and lycopene together may be better. Phytoin alone and lycopene reduce the risk, but the bottom, the blue line shows the combination of phytoin and lycopene, which is synergistic. This may be because there are different mechanisms that are induced by different carotenoids. For example, beta-carotene is much better in inducing retinoic acid response, whereas lycopene is better in the PPAR gamma response. So when you're ingesting the beta-carotene and lycopene together, you're basically taking advantage of different mechanisms, and the combination therefore may be working better. So after we saw these results from epidemiologic studies and in vitro studies, we conducted a clinical trial at Wayne State University in Detroit. We took patients with prostate cancer who were getting ready to have their prostates removed. These are patients with prostate cancer. And we gave them 3 weeks of lycopene, and there was a control group with no lycopene. And this is the product that we gave them. We used a nutritional supplement product called Lycomato, which was made of lycopene, mostly lycopene, but it had all the other carotenoids found in tomatoes because this was a natural tomato extract. And when we looked at the patient's prostate, when we took out the prostate, we were able to find all the carotenoids that are found in the tomato plant. So we were able to show that actually what they consume when patients take the lycopene capsules, the contents of the capsule actually end up in the prostate. So we were able to show that on the chromatogram from prostate tissue. We also looked at some clinical parameters in the lycopene group. There was more disease confined to the prostate. Even within 3 weeks, maybe we were able to see some shrinkage in the tumors. The tumor volume also less in the lycopene group. The numbers are small, so the p-values are not significant, but you see the trend. In the lycopene group, the PSA level went down. In the control group, it went up as expected. And also pre-neoplastic lesions in the lycopene group. There were more focal lesions where there was none in the control group. We thought one of the mechanisms may be connexin 43 because connexin 43 gap junctional protein is known to be upregulated by lycopene. And indeed, in the prostatic tumors that we removed, these are very small numbers. Of course, there are only 4 patients from the lycopene group, 4 from the control group. We can still see the trend that connexin 43 level is increased. There was no significant changes in the other factors that we looked at, like BCL2 or BAX. In the benign part of the tissue, we were able to show that lycopene level did increase after we gave those capsules to the patients. So we concluded from that study that lycopene supplementation 3 weeks is well tolerated, and we didn't see any significant side effects. And the connexin 43 was upregulated, and some of the clinical markers were improved in the right direction. And we published these results in Cancer Epidemiology, Biomarkers, and Prevention. And unbeknownst to us, it's totally unrelated. Another group in Chicago was doing the same studies, and they published it a month after we did, almost identical results. So we were able to see, they published in JNCI, almost identical results. So we were able to confirm in 2 separate studies that are totally done independently the same kind of results. So this was very encouraging. So after that, a lot of patients started asking us about lycopene. Patients who had surgery and radiation, and now who have rising PSAs, which means the cancer is coming back, but it's not metastatic yet. Some of these patients don't want to take hormone therapy, which is a standard therapy. They want to try something else before going to hormones. So I have over 100 patients like that who take lycopene over the counter as a supplement, and they're able to keep their PSAs under control. This is one example, a cardiothoracic surgeon who's been on lycopene for more than 11 years when he came with elevated PSA level. So like Hippocrates said, food can be medicine. Another thing that I would like to talk about is soy isoflavones. That's my second area of interest. Diet and fatal prostate cancer. This is one example of how dietary factors can have bad effects. Dairy products have been associated with worse prostate cancer. However, drinking soy milk has been associated just the opposite, decreased risk of prostate cancer. So soy isoflavones, genistein is shown here on this figure, which is a prototypic soy isoflavone, which has similar effects to tamoxifen, which is a drug used for breast cancer prevention and treatment. It binds preferential to estrogen receptor beta, and they bind to the same receptor estradiol binds, but preferential to beta, so they're selective estrogen receptor modulators. But genistein has other effects too, not just through the estrogen receptor, but it inhibits rank ligand, promotes vitamin D activity, inhibits inflammatory molecules like IL-1, IL-6, TNF-alpha, and NF-kappa B. So given the soy isoflavones have been associated with low risk of cancer in epidemiologic studies, we wanted to look into that a little further. And we did some in vitro studies looking at genistein in prostate cancer proliferation. We were able to see a decreased, these PC3 cells are aggressive prostate cancer cells. They are castration resistant. We also looked at improvement in chemotherapy efficacy in prostate cancer when we used genistein together with chemotherapy. We were able to see a synergistic effect. So we looked at why this may be happening. With chemotherapy, NF-kappa B is activated, and that confers resistance to chemotherapy. But when genistein is used together with chemotherapy, there is no upregulation of NF-kappa B. So that we think is one of the mechanisms why giving soy products like genistein may improve the efficacy of chemotherapy. And this figure shows this hypothetical mechanism, how genistein, by inhibiting NF-kappa B, can also inhibit other molecules downstream that are important in cell growth and metastasis. We also looked at in animal models, genistein in prostate cancer metastasis, and we were able to show that genistein has anti-metastatic activity in prostate cancer. We were able to also show the same thing, that improvement in chemotherapy activity with genistein, as you can see in the left side, when genistein and chemotherapy are given together, tumors are much smaller in the animal model too. We also looked at gene expression profiles from animals that are treated with genistein. These prostate cancer tumors, they have very high metalloprotease activity, for example, seen here in this picture. And when genistein is given, the expression of matrix metalloproteases decrease. We looked at a number of other molecules that are the categories of different molecules. The different genes are shown here. Many genes are upregulated or downregulated with genistein, which has significant effects on gene expression profiles. Genes that are involved in cell growth are decreased, and those that are involved in apoptosis are increased. We also looked at Wnt pathway inhibitory genes, which are hypermethylated in prostate cancer patients. This is from our clinic. These patients, we picked and looked at their gene expression profiles. These are the genes listed on the left-hand side that shows that genes are hypermethylated. And when we looked at the whole genome profile of these tumors, RCAP-M and RCAP-E. RCAP-E is the epithelial type. RCAP-M is the mesenchymal type, which is a worse type of tumor. You can see there are significant changes when they're exposed to genistein. Many genes are downregulated and upregulated, shown as in this heat map. And when you look at the gene expression profiles, genes that are involved in cell cycle responses to DNA damage are increased, and genes are upregulated that are involved in cell cycle response to DNA damage. And genes that are involved in TNF, NF-kappa B pathway are decreased. So we also did a clinical trial. We took patients with rising PSAs. G3 and G2 are two groups of patients. One group is never received hormone therapy, one group with hormone therapy. And you can see in both groups, after the patients start on taking soy isoflavones, the curve starts flattening. So in other words, when the PSA is doubling every 3 months, now it's doubling every 6 months. So we are able to give patients more time without the toxicity of hormone therapy and chemotherapy. So in summary, genistein and soy isoflavones, they have many mechanisms of action which may be important in cancer prevention and treatment. They have antioxidant effects, so preventing DNA damage. They may explain some of the reasons why they prevent cancer risk. They have anti-inflammatory effects. As we all know, many of the cancers are associated with increased inflammation. There are also epigenetic effects of genistein through DNA demethylation and histone acetylation. I didn't show you the data, but we also showed increased histone acetylation, inhibition of NF-kappa B, rank ligand, VEGF, MMP, EMT. We've published these results. So we have also seen in preliminary studies, there is increased chemotherapy and radiation therapy activity, better results when genistein is added to chemotherapy and radiation. We have also observed decreased toxicities of chemotherapy and radiation therapy because the toxicities of chemo and radiation may be associated with inflammation, and genistein as an anti-inflammatory may benefit that. I haven't shown you the data. There are also data showing it improves immune function, and it has also antiviral and antibacterial effects. Again, these are results I didn't show you, but obviously these are very important in patients with cancer who frequently have infections, especially when they're getting chemotherapy. So like as Hippocrates said 2,000 years ago, let food be your medicine. There are a lot of botanical compounds. There are many that I haven't mentioned. I just mentioned 2 of them in this presentation, but there are green tea polyphenols and many products in nature, and a lot of them are in our diet. They're part of our diet, and they're very easy to take in biologically active quantities. It's very easy to get enough lycopene or soy isoflavones just by consuming a diet high in soy products or tomato products. So I will stop here and answer if there are any questions. Thank you.
Thank you. Really a very interesting presentation, and I hope that we can continue changing ideas, and probably we can do a Zoom meeting in the future to look for ways to cooperate. This is very interesting. I think there is no question in, oh, there is some questions, yes, here.
Hi, thank you very much for your very nice presentation. But prostate cancer, like other cancers, are multifocal in origin, implying multi-clonality. So I was wondering whether either lycopene or genistein or any flavonoids would impact on the clonality, or in other words, whether they would reduce the number of clones or they would just reduce the growth of the entire multifocal tumor. That's one question. Second question is, from the properties you mentioned about for genistein, for example, could it also affect aging, not just cancer? For example, it's anti-inflammatory, and it has all these other properties. Could be also a factor or a compound that could impact or benefit aging or reduce unhealthy aging. Thank you.
Yes, these are excellent questions. Thank you very much. As far as clonality, of course, not every cell will have the same kind of response. So different clones may have different responses to genistein and lycopene. And when tumors get more aggressive, when they progress into a chemo-resistant stage or hormone-resistant stage, they become less responsive to treatments, not just standard treatments. For example, in prostate cancer, we use hormone therapy and chemotherapy and radiation. These are standard treatments, and tumors get more resistant after they are treated with hormone therapy and chemotherapy. Almost nothing will affect them. So obviously, things like lycopene and genistein will be of very little benefit in these very resistant clones. But when patients first develop their disease, they usually have a very mild type of disease. They start out with more indolent disease, and they're more susceptible to nutritional manipulations, nutritional interventions. These may have more benefit in the prevention setting rather than after cancer develops. After cancer develops, they may have a role as an adjunct to standard therapy. But many studies show that, I've shown those studies related to lycopene. There are also a lot of studies with soy. Women, for example, who consume soy products, they have a much lower risk of breast cancer. And women who have breast cancer, who continue to consume soy, they do better. They live longer. They have less recurrence. So there are certainly benefits of consuming soy products before and after developing cancer. And as far as the aging, yes, there are also data that shows that for healthy aging, soy products and other nutritional products like tomato products and vegetables and a healthy diet is very important for healthy aging. An anti-inflammatory diet or antioxidant diet may be very important in prevention of cancer and also in having a healthier aging with less diabetes, less hypertension, less cancer, and less chronic diseases. And even there is some data that Alzheimer's may also, brain function, cognitive function may also be improved with these agents.
Thank you very much. And I think that's all for today. Thank you, Professor Kucuk.
Thank you.
And now we are going to have a break, so we will convene back at 11 a.m. to our audience who is following us. Thank you.
Thank you.
Hello, hi, everybody. We are back. We have now with us Dr. Tanya Grkovic. She is an staff scientist at the Natural Product Branch of the National Cancer Institute. Dr. Grkovic is a natural product chemist, focuses on identifying and understanding biological active compounds. Her work involved isolated natural products from marine and plant sources, elucidating their structures and contributing to drug discovery effort through high throughput screening. Welcome, Dr. Grkovic.
Thank you very much. I'm about to share screen. Let me set up my Zoom. And do you see my screen?
You need to put yes, we do. Yeah, the full screen. Yes, perfect.
Okay, great. Thank you for the introduction, and many thanks to the conference organizers for the invite. I am very happy to be here and present some of the work that we're doing at the NCI on natural products. So my intro slide, this has been said before, natural products have been and continue to be major sources of drugs and drug leads in the clinic. Over 30% of all small molecule-based drugs are natural product derived, and in the last 40 years, natural products and their derivatives have represented 50% of antibacterial drugs, 45% of antiparasitic drugs, and 40% of anti-cancer drugs. However, when we have a look at PubMed citations and high-throughput screening, we see that it's had an exponential increase from early 2000s to about 2010, and now plateaus at about 2,000 publications a year. But when we cross-reference that with natural products, we see that natural products stay the baseline. So we have this amazing treasure trove of drug potential that's just not getting screened using modern high-throughput targeted screening technologies. And why? There are many issues. These are just some of them. So solubility is one. Natural products are complex mixtures of primary and secondary. Natural product extracts are complex mixtures of primary and secondary metabolites. There's anywhere between 100 to 1,000 compounds in an extract. The image on the left, the 96 well plate, is what our screening lab calls the plate from hell. And you can easily see why, because there's solubility issues, there's stuff crashing out. There's also a lot of different colors that may interfere with assay endpoints. Cytotoxicity and off-target issues is another big issue. So the image in the middle shows a kinase assay straight out of the kit, screening this natural product library unoptimized. Is going to give a gigantic hit rate. Here we're showing a 40% hit rate on an unoptimized assay. This can be approved. So the green graph shows a hit rate of 0.3%, but it takes a lot of methods development to get it there. And then finally, the last point is there's lengthy isolation procedures. So natural products, complex mixtures, it takes several steps of isolations before we even get to the structure. So usually in an HTS campaign screening crude natural product extracts, it's quite often that an HTS is over before the structure is determined. And that's a big problem. The NCI is home to the Natural Products Repository. It's one of the largest collections of biota used for natural products-based research. It's also incredibly diverse. So our collection houses over 200,000 extracts, mainly from plants. It's represented by over 5,000 unique plant genera, over 3,000 unique marine genera, and just under 1,000 unique fungal genera. However, in its current form, this resource was crude extracts only. And I've shown in the last two slides the challenges and problems associated with crude extracts. So about 10 years ago, the NCI started a new initiative called the NCI's Program for Natural Products Discovery. And there were many aims, but the main aim was to modernize the repository and make it more friendly to high throughput screening. So the initiative was based on the prefractionated library. And so the main aim was to prefractionate the repository and generate a library of 1 million natural product fractions for modern high-throughput targeted screening technologies. This library is publicly accessible, free of charge, and open to screening against any disease target. And in addition to generating the library, we also wanted to create analytical methods and analytical chemistry resources and methods for screening support. So central to this is the library. Our prefractionated library is made with prefractionating, taking 250 milligrams of the crude extract and prefractionating on C8 solid support. We're using a 7-step methodology where we start with 95% water, 5% methanol, and then climb in methanol, 20, 40, 60, 80, 100% methanol. And the last solution is the column wash, which is methanol acetonitrile mixture 1:1. Because our numbers are so big and the aim was so ambitious, we had to automate. So what you're looking at the bottom, and I'm about to start the video, is a TCAN liquid handler that's been completely customized to perform positive pressure chromatography. So we're going to start with equilibrating columns. Then we're going to add our crude extracts in, and now we start the prefractionation. So an instrument like this with 2 arms working in tandem can prefractionate 88 extracts in a single run to generate 616 fractions. Running 4 runs a week can generate just under 2,500 fractions, and running 50 weeks a year can generate 125,000 fractions. So now all of a sudden, this goal of making a library of 1 million fractions isn't so scary anymore. So fully automated methodology was developed. This extract, like we mentioned, was separated into 7 fractions. So far, we've made 650,000 fractions. We sequentially released them every couple of years. We're due for a new release next year. Currently, there's 500,000 fractions available for HTS. This is how the library for now looks like. So the majority is plants with 86,500 fractions sourced from the marine environment. The fractions are plated on 384 well plates containing 10 micrograms per well of individual samples. So we plate both the fraction and the crude extracts, and they're dissolved in 2 microliters of DMSO. It's not a lot, but it's plenty to support a couple of different high-throughput screens in each plate. We opened the library for screening against any disease target. However, access to the library requires a signed MTA that acknowledges the intellectual property rights in the source country of the biota collection. Having screened this library in multiple screening campaigns over the last couple of years, and having the benefit of both the crude extract and the fraction on plates, we can see that in about 70 to 80% of the time, activity was identified in the fraction, but not in the crude extract. And that really, really confirms the value of prefractionating, right? We're concentrating these minor compounds that we would have missed if we were screening just crude extract libraries. So that's the first step, screening the library. During methods development, we had a look at how many compounds are in each fraction. So this is now a simplified natural product sample, but it's still a mixture. The real answer is depends on which pairs of eyes you look at it with. So when we look at it with the eyes of the mass spec and ELSD, there's an average of about 25 compounds in each one of our fractions. So much simpler extract, but what we really want to do is we wanted to make methodologies for the next step where we go from this simple mixture to pure or semi-purified compounds. So in our screening workflow, the next step is something we call second stage HPLC, where we take 1 milligram of the fraction material, put it on a semi-preparative column, and then collect 22 subfractions by time. We're capable, so we built methodologies where we're capable of taking 500 primary hit fractions, doing this second stage HPLC, subdividing into 22 subfractions, and returning it back to the screening lab within 2 weeks so that the cells and the assay is not waiting. In addition, at this point, we start getting analytical data on our hits. So the idea is we're going to generate these second stage plates, send it back to the screening lab. When we get the results back, we're going to start looking at the active wells and only the active wells. And we're going to get as much information as we can on them. So we start with proton NMR, get HSQ, see if the signal to noise is adequate. We also collect MS1 and MS2 data, as well as FTIR. So the idea is get as much as you can so that we can start dereplicating and introducing structure. So this methodology enables us to introduce knowledge of structure early in the isolation workflow. So only 2 automated steps. And it significantly reduces the cost of natural product screening and creates timelines more amendable to high throughput screening programs. So basically, it enables the speed of natural products to match the speed of high throughput screening. How we're doing so far, there's 500,000 natural product samples publicly released. Over the last 10 years, we shipped 9 million wells to screening centers worldwide. Our shipping locations in the continental US are shown on the image on bottom left. We've done technology transfer and methods development to labs in South Africa, Sweden, and the US. And we've signed over 70 MTAs with industrial, government, and academic screening partners. So the library is getting screened. And for the rest of my talk, I'm going to show you an example of a high throughput screen of our library that basically goes back to all of these ideas that we've covered here. So this was an antimicrobial high throughput discovery campaign. It was a collaboration between the National Cancer Institute and another institute of the NIH, NIAID, which is the National Institute of Allergy and Infectious Diseases. The value proposition of this screen was to take 4 different targets, screen the whole library, and then make the results publicly available. The hope was that this is going to encourage further interest into screening natural products for antimicrobial targets. So what we had was gram-positive, gram-negative bacterium and a fungus. The single-dose screen was done at 10 micrograms per mil in duplicates. The hit rate was big because the hit rate definition was really, really broad. So anything with an inhibition value of 4 standard deviations within the mean value of the 50% of the replicates was considered a hit in this initial screen. So anything that's colored basically in this graph on the right. It gave us a really, really big hit rate of 2.9%, but that was okay because our reconfirmation, the next step was going to be a dose response screening. So what we really wanted to do here is we just wanted to cast a really, really wide net of activities that we can reconfirm later. The dose response was done at 8 doses, starting from 0.08 micrograms per mil to 10 micrograms per mil. And here the hit rate was a more responsible 0.9%, which if you consider it's got 4 targets, it's actually really good. Most of our hits were antifungals against C. albicans, followed by S. aureus. These are the violin plots looking at. So most of the hits were centered between 10 and 5 micrograms per mil, but we're also showing a significant amount of hits that go to low micrograms per mil or even nanogram per mil ranges. Here's a couple of examples. So the one on the left shows a non-selective hit that hit all 4 targets. Clathrina, Darwini in the middle shows an antifungal hit. And then the last 2 are hit that shows activity against E. coli and S. aureus. They're from the same genus of sponge, but the first one is a non-polar hit. So subfraction, I need to get to my laser pointer. So both of these come from the same genus of sponge, but this hit comes from fraction 7, which is non-polar and is showing activity against E. coli. I'm going to say it's not the actual same specimen of a sponge. It's just 2 different collections of the same sponge. Whereas one of them is more polar, fraction 2, that elutes with 20% methanol, 80% water, and it showed activity against S. aureus. I'm taking it further. So we wanted to show proof of concept. So what we did was we took 75 of those hits, and we wanted to show sort of how do you go from simplified mixture to pure or semi-purified compounds, and how we usually treat our HTS screening campaigns. So we chosen 75 hits that were based on broad spectrum of potency. We didn't choose the most active. We chose a broad spectrum of all of those violin plots. We had a look at other NP-MPD assays, and we've chosen hits that showed no activity in other assays or lack of cytotoxicity against cancer cell lines. And then we've also made sure to incorporate taxonomic redundancy by choosing only a single specimen of each genus. And this really is, when we do high throughput screening, this is our basic selection funnel for choosing hits for further work on. So here's an example. Then after choosing these 75 hits, we took it to second stage HPLC, and what you're looking at is an actual example of the second stage HPLC from this Ascidian. So all of the peaks that show the UV profile were actually active. So this is a heat map of activity. It shows selective and potent activity. And then we started getting analytical data. So on the 75 hits after screening, there was about 400 samples that we recorded: proton, NMR, LCMS, and IR. And we do a lot of metabolomics to try and dereplicate the hits. So this is a summary of a whole lot of work. What you're looking at here is the mean cluster assignment of the proton NMR data that basically looks at grouping these hits according to similarity of NMR data. This is the self-organizing map of FTIR data that pretty much functions the same. And then finally, the LCMS2 molecular network. In addition to doing a lot of metabolomics, we also look at all of these spectra. We match them against databases of known compounds so that we can assign a structure or chemotype to each hit. This is the results of the dereplication study. And this really helps us. Using this and working on just 1 milligram of the fraction material, we were able to assign chemotypes to about 70% of the hits. That saves a lot of time and effort in the chemistry lab and enables us to pick good hits. So for example, some of these hits like the polyacetylenes, the plant tannins, or the marine saponins are considered nuisance compounds. And we didn't have to use time, effort, and the extract to actually get to this. We only use 1 milligram of the fraction. And it enables us to choose hits that have new interesting chemistry or interesting biology. So here we took a couple of these projects. I'm going to show the results of 2 marine projects that we took forward. So 2 of the sponges, a Lucetta and a Clathrina sponge, was my example of selective antifungal activity earlier. We found that both of these have the same chemotype. They made these 2 aminoimidazole compounds. We've isolated 3 compounds. And when we tested them against our microbial panel, we found that they show potent and selective antifungal activity that hadn't been reported in the literature before. Another example that showed S. aureus activity was a Petrosia species sponge. The active principles were found to be pyridoacridine compounds. With these deoxyamethidines, compounds 4, 5, and 6, showing potent and selective activity against S. aureus that, again, was not reported before in the literature. In conclusion, the NIAID study identified 3,000 fractions with antimicrobial activity. We demonstrated fast and effective active principal isolation protocol, and we identified new natural products. The results of this HTS were made publicly available for anybody to have access to. And that's it. I'm going to thank, again, the organizers for inviting us to give a presentation, and I'm going to thank you for your attention.
Thank you very much, Tanya. Any questions from the audience? No, it seems like we do not have a question right now, Tanya. So thank you very much for your comprehensive presentation.
Thank you.
And now we have with us Dr. Antonio Currais. Is Antonio here? Antonio?
Yeah, yes, I am. Can you hear me?
Welcome, Antonio. I will say something about you before. Dr. Antonio Currais is a staff scientist from the Salk Institute for Biological Studies. He focuses on understanding aging-related neurological diseases. His effort contributed to preclinical studies on drug candidates which target the aging process in Alzheimer's disease. The floor is yours.
All right. Thank you, Veronica. Can you see my presentation?
I also put it in full screen.
Okay. All right. Okay, so thank you, Veronica, and thank you for the invitation to present some of our work today. It's always a pleasure. Some of you might have already heard a few of these things, so I apologize. For the others, I hope you enjoy it. So I'm going to be very briefly talk about how we've been developing therapeutics for treating dementia with a focus on Alzheimer's disease. We've studied other neurodegenerative diseases in the past, but our our main disease is Alzheimer's. So this is a photo of our institute. So in our lab, the cellular neurobiology lab, we do both fundamental research as well as drug discovery. And the idea is that the knowledge that we acquire by studying basic processes underlying aging and dementia will allow us to design better assays that we then apply to identify and develop therapeutics. And because of the way we do this, as I will explain, and I hope it will be clear by the end of the presentation, we also end up understanding how the disease works. So as probably many of you know, there really are no effective treatments for dementia, for Alzheimer's disease. And we think that part of the reason is because most of the research has been done around what it's called the amyloid cascade hypothesis. So basically, this hypothesis postulates that the deposits of extracellular amyloid beta plaques in the brains of Alzheimer's disease patients, which you can see in the photo on the left, are the culprit of the disease. So they are the cause of the rest of the pathological hallmarks, including the neurofibrillary tangles of another protein called tau, which eventually leads to the neuronal cell death and the manifestation of the disease. However, this hypothesis has many limitations, even with the recent amyloid immunotherapy, which I'm not going to get into that. And what we have been trying to do is to think what could be even upstream of this. And we know that aging is the biggest risk factor for Alzheimer's disease. The probability of developing the disease increases exponentially as we get old. And we know that aging is a complex process that is characterized by a deterioration in many different systems in the cell. Some of these are found altered in the aging brain and exacerbated in Alzheimer's disease. And so this has basically led us to our working hypothesis that by targeting specific aging processes that we still need to identify, we may be able to prevent early events that lead to dementia. So how do we do this? So we historically, the lab started even before I moved to San Diego. The lab was already working with natural compounds and synthetic compounds, pure compounds. And eventually, after I moved there, we started becoming interested in also working with extracts. But that was just my own interest. But basically, so we start with libraries of natural compounds or natural product extracts that we then test in cell-based phenotypic screening assays, which I will explain in a minute, to basically identify compounds that are neuroprotective in these assays. Then these compounds can be further optimized chemically to improve the potency in the screening assays, improve their safety, their pharmacokinetics. And then once we're happy with candidate compounds, then we test them in animal models. And animal models for Alzheimer's disease has also been a huge challenge. And I will briefly tell you how what is our approach. But basically, because of the way that we do this, we end up understanding not just how compounds work in the animals, but also identifying how they work, so the mechanism of action. And this allows us to advance drug candidates for the disease, to identify novel therapeutic targets, and also to provide a basic understanding of the disease. So what sources of natural products, natural compounds do we use? So we have shown that we can use many different sources. So we've worked with libraries of pure natural compounds. So in this study, we basically used a commercially available library of over 1,000 natural compounds from different biological organisms. And actually, in this study, we show that for one specific assay, which I will tell you in a minute, plants seem to be a very good source. But that's probably varies a lot. We've also shown that we can use libraries of plant extracts, of crude extracts. So this was a study where we basically used also commercially available library with hundreds of different plant extracts. And it was very interesting because we, through bioactivity guided fractionation, ended up identifying the active compound, and it turned out to be a sterubin from a plant that is actually native from California called Eriodictyum californicum. All of the library had plants from all over the world. And we also work together with people with labs that work on ethnopharmacology or plant biology. Okay, so how do we proceed with the phenotypic screening assay? So throughout the years, we have established cell-based assays that basically mimic different toxicity pathways that we think are relevant for aging and also for Alzheimer's disease. And some of these assays, so they basically, they're all cell-based assays. This is the first layer of our drug discovery approach. And most of them can be carried out using 96-well plates. We don't do high throughput screening in our lab, so everything is manual. But we can test a lot of compounds or extracts relatively quickly. So we have assays to mimic growth factor loss, and we use primary neurons for this. We have another assay that basically exposes cells to an endogenous source of oxidative stress called oxytosis, which is also known as ferroptosis. So it's not exogenous. The cells are not treated with an exogenous pro-oxidant. They are made through inhibition of specific signaling pathways. They are made to go into this non-apoptotic cell death process where there is a burst of oxidative stress, mitochondrial dysfunction, and it basically ends up with a chaotic lipid peroxidation event where the cells die. And it turned out to actually become the main assay in our lab, and also the main focus, as I will tell you in a minute, of our basic research. We also have an assay for energy loss. It's basically in vitro ischemia, where nerve cells are treated with inhibitor of glycolysis. So in many of these, the outcome is cell death, right? So we give the insult, the cells die, we treat them with the natural compounds or the extracts, and then we assess cell survival. We have also an assay for where we treat cells with extracellular or intracellular amyloid beta toxicity. And then we have an assay for inflammation. In this case, we use a microglial cell line, and the outcome here is basically the production of nitric oxide. So cells are stimulated with lipopolysaccharide to simulate an infection, and then we basically see whether the compounds can tame the inflammatory response. And then we measure nitric oxide, but we can also measure a bunch of different cytokines. We have another assay for nerve cell differentiation, where cells are treated cells can develop neurites, and we can measure that. So these are basically all for the nervous system. And more recently, we have been adding additional assays, and one of them regards senescent fibroblasts. And the reason why we do this is because there's been more and more interest in understanding how the periphery affects the brain, namely with aging, because aging likely takes place at a systemic level. So that probably may contribute to the development of Alzheimer's disease. So we have introduced an assay for cellular senescence. Basically, fibroblasts are made replicative senescent, and then we can assess the effects of compounds and natural product extracts on the replicative senescence, as well as on the production of the SASP, which is the secretory associated senescent phenotype. So it's basically these cells, these fibroblasts, once they become senescent, and that happens as we get old, they go into this senescent state characterized by metabolic shift that leads to cell cycle arrest and the production of pro-inflammatory cytokines. So we can measure those. And we're also, right now, we're thinking about implementing another assay that is based on the generation of induced neurons through direct reprogramming using fibroblasts from older people. So basically, the idea is that, or used to be when we started doing this, that we wanted compounds that protected against all of these assays, or as many as possible, because if aging and the disease is characterized by all of these things, we want to target all these processes, right? And so once we have a compound that we're happy with, then we put them in animal models. And just like many other people in the field, we also work with mouse models transgenic mouse models of Alzheimer's disease. So most of these mouse models basically have what it's called familial Alzheimer's disease mutations. So these are specific genetic mutations that are known to lead to the early development of Alzheimer's disease. However, familial Alzheimer's disease only accounts for maybe 3% of all the cases. The majority, it's called sporadic Alzheimer's disease with onset at 65 years old. And there's no model for that. And this has been a huge limitation. And because of this, that's part of the reason why we started looking into the aging process. So we've introduced also mouse models of accelerated aging that they develop features relevant to dementia that we use in the lab. And we work in collaboration with labs in San Diego that use other models such as the worms and flies. And basically, we do a really comprehensive set of analyses looking at cognitive behavior. We also look at large-scale analyses, including the transcriptome, the metabolome, and the proteome. And together with the bioinformatics core at Salk, we developed a set of tools that allow us to integrate all this data, identify candidate molecular pathways that then we can validate back in cell culture or in the animal models. And I'm just going to give you an example of how this worked out for two of our most potent drug candidates. One is called CMS-121, the other one is called J147. So these are both derivatives of natural compounds, CMS-121 from physatin, a flavonoid, and J147 from curcumin. So very distinct structures. But basically, so these compounds were initially selected using the phenotypic screening platform that I explained to you before. And using the multiomics analysis that we're using right now, we're able to show that both compounds were able to prevent the development of aging in our model, and they actually seem to do that through similar mechanism of action that involve the triggering of an energetic stress response mediated by AMP kinase. So AMP kinase is a sensor molecule enzyme in the cell that can become activated, phosphorylated, when there is stress in the energy levels. There are also other causes that can activate it. And it has been linked to prolongevity, activation of prolongevity pathways. So, but basically, we've shown that through activation of AMP kinase, that leads to an increase in a very important metabolite that is found in mitochondria called acetyl-CoA. And so that's how we realized that the compounds could be maintaining mitochondria health with aging. So at the same time that we were doing this, we had two postdocs trying to figure out the molecular targets of these two compounds. And because this is a phenotypic screening assay, we didn't know their targets, right? And one of the reasons why we do phenotypic screening with Alzheimer's is because we don't have good targets, or there is a lack of targets other than the amyloid and tau pathology. And because also we don't know what aging is exactly. So we work with the phenotypes, and then we try to understand how these drugs work. And many times this helps us finding new targets and understanding the basic biology, right? So in this study, my colleague Josh found that J147 was a modulator of the mitochondrial ATP synthase. So this was really interesting because the mitochondrial ATP synthase makes energy, most of the energy in cells. And he showed that basically J147 binds to the mitochondrial ATP synthase, and it's has been shown that it can extend the lifespan in wild-type mice, flies, and worms. And in another study, Gamza found that CMS121 inhibits the fatty acid synthase, which is an enzyme that is involved in fatty acid synthesis. And he did that. This was shown in Alzheimer's disease transgenic mouse model. He did that along with reducing levels of lipid peroxidation and inflammation. So, but basically, the interesting thing is that two different compounds that in structure that were selected using the same phenotypic screening assay actually have two molecular targets that fall on the same molecular signaling pathway. And so we've shown that both of them activate AMP kinase. And we've also shown that AMP kinase activation, what it's doing is inhibiting an enzyme called acetyl-CoA carboxylase 1, which is a rate-limiting enzyme involved in the conversion of acetyl-CoA into malonyl-CoA, which is used as a precursor of fatty acid synthesis. So free fatty acids in the cell are very susceptible to oxidation, which can happen through lipoxygenases or reactive oxygen species. And if that reaction is uncontrolled, we can have intense lipid peroxidation with what is called oxytosis ferroptosis, so which is one of the screening assays that we use. So we've shown this by genetic and chemical manipulation of all these molecules. And this is how we think these compounds may be basically preventing the contribution of aging to the disease. At least that's our hypothesis, what is also called neuroprotection. So, summarizing, basically, I've shown you that the approach that we have in the lab allows us to develop drug candidates for Alzheimer's disease. And both compounds have already gone through a phase 1 clinical trial. We're trying to get funding to have a phase 2 with CMS121. We also identify novel therapeutic targets. It's called the chemical biology approach, as is the case of the mitochondrial ATP synthase and the fatty acid synthase. And so this approach also allows us to provide fundamental understanding of what are the mechanisms operating during aging and Alzheimer's disease, such as identification of oxytosis ferroptosis, which we are now pursuing in the lab. So I want to thank you for your attention. And this is the rest of the lab. It's already an old picture. And some of these people are not around us anymore, unfortunately. So thank you.
Thank you, Antonio. Excellent presentation. And here we have a question.
Do I stop sharing so that I can see you, or?
I don't know, is any way that he can see me?
I'm going to stop sharing and then I can share again if.
Here is the question.
Thank you for the great talk. Recently, they've discovered that misregulation, epigenetic misregulation of retrotransposable elements leads to aging. My question is, do your assays look at that in the transcriptome area?
No, we do not actually.
And then I guess a follow-up question, have you noticed any compounds be retrovirals that actually impact the disease?
So we've never screened specifically retroviral, antiretro, yeah, so.
Yes, you did. Yeah. Here is a second question.
Yeah, hi. Yeah, also increasing acetyl coenzyme A, would it increase histone acetylation? I think it might also affect epigenetic and otherwise, transcription regulation. Have you looked into that? Yeah, histone acetylation, for example, or
yes. Well, that's a very good question. Yes. So we did see in the original paper, we looked into protein acetylation. The drugs actually increase total global acetylation because acetylation can happen non-enzymatically as well. We actually saw that the first increase, or at least part of the increase, happens inside the mitochondria because we isolate the mitochondria and we probe for pan-acetylation, right? And the compounds increase it. But we've also seen acetylation of specific histones, namely H3K9, which has been reported to be involved in memory, cognition. And right now, we are working under a grant, basically with the purpose of identifying the mechanism that leads to the epigenetic changes underlying the action of one of the compounds, J147. So we are actually right now doing that. And we identified one of the transcription factors, yeah.
Thank you very much, Antonio. I think we don't have any more questions now, yeah? ¿Cuál es la pregunta? Yeah.
Hi, Antonio. We have one last question from our online public. It's from Selene Manga, and she asked if the antioxidant power of certain plants used for altitude sickness, such as cola leaf, will have an impact on brain anti-aging.
Sorry, do you mind to repeat the question again?
She asks if the antioxidant power in certain plants used for altitude sickness, like I believe it's coca leaf, will have an impact on brain anti-aging.
So we never tried coca extract. The antioxidant question is very common, and I don't personally. I'm very skeptical of studies, especially in vitro studies that just look at the antioxidant activity of compounds, because what we see is that actually there is no correlation between antioxidant activity. And there are many assays to measure different types of antioxidant activity in vitro. There's really no correlation between that and the protection against oxytosis ferroptosis, which is a pathway where we induce endogenous oxidative stress. Although some antioxidants do protect in that assay. But I think part of the problem is related with the fact that you need to pay attention to other aspects of the structure of these compounds. Anyway, I think Veronica is already looking at the watch. But anyway, so we've never looked at coca extracts, yeah.
We can continue the conversation with Selene and Antonio in the future. Yeah, we can
All right.
have discussion. That will be interesting. Okay, thank you, Antonio.
Thank you.
Yeah, and now we invite Miguel Angel Chavez to present. Miguel Angel is a full-time researcher at the Catholic University of Santa Maria in Peru, where he is the head of the Computational Biology and Chemistry Research Group. His research interests are applying bioinformatics to discover and develop drug, diagnostic, and vaccines against neglected tropical diseases. Welcome, Miguel.
Hi, can you hear me? Oh, you had to start. Okay, thanks. Well, I want to thank the organizers for inviting me. This is the first time I participate in this seminar, so I do appreciate a lot for the opportunity to first time being in Costa Rica. So for me, it's a special time. So as they present me, I'm Miguel. I work at the Catholic University of Santa Maria at South Peru, and I am the head of the Computational Biology and Chemistry Research Group. So I'm gonna talk about part of the research we are doing at the research group. So I think we all love Newman and Craig review. I think it's the third or fourth time we see it here in the seminar, but just to see how important is on research and drug discovery. So Peru, it's one of the mega diverse countries in the world. I think there were 17 only among the almost 200 countries, and we have a traditional use of more than 5,000 plants of traditional use. So there is a good knowledge of the use of plants, and we have a lot of biodiversity, but it is only being translated to a drug in one time, which is quinine for the treatment of malaria. So we have a lack of research among Peruvian natural products. When I came back, when I returned from Brazil to Peru nope. When I came back from Brazil to Peru, one of the main ideas was to try to develop some work on the field. So in 2020, burns the idea of the implementation of the Peruvian natural products database, which gathered with the aim is to gather information and some of the compounds that were derived or okay.
So it's better, no? Yeah. So I can just because I don't really remember what is the things. So we start at 2020 to gather the information on the natural products. So we got in 2023 the publication of the natural of the Peru NPDB, and we got it online. So it's a database free to use, and you can. I think we are the only one. We have a application for Android,
and we are maybe in the second version, we are going to have it on iOS. So if you want to take a look on the paper, you just you can go to the QR code, and if you want to take a look on the database, you can take a look on the QR. So we started by looking at and reviewing almost 400 papers, and we got at the final filters, we got 280 compounds. And one of the characteristics we observed is that almost all the compounds were from plants, and only 5% for animals. We didn't found other organisms information like fungi or microorganisms like bacteria, or other sources. We didn't found any study with these sources. So we characterize 37 genera and 76 classes of compounds. When we studied the physicochemical characteristics of the compounds, we use other three databases: Nube, which is from Brazil, Biofakim, which is from Mexico, and AfroDB, which is a database which compiles the information from different countries of Africa. So we saw that there is some difference between the databases, and we analyze the distribution on the chemical space, which is these fancy graphs. We see that it almost overlaps with some difference, but it overlaps finally. So in the last 4 graphs, we since it's an idea to have it for virtual screening for drug discovery, we compare it to the data set of the drugs approved by the FDA. So we saw that it has overlapping at the chemical space, and the number of violation of Olympic values were very similar. So it can be employed in campaigns for virtual screening for drug discovery. But one of the finding of the paper was when we analyzed the chemical diversity of the data set. When compared with the other 3 databases, we saw that besides is the smallest data set, we saw that its molecular properties were the most diverse of all the data sets analyzed. So it was this was already seen in other studies, like when we participate on the construction of the LANA PDB, which is the Latin American Natural Products Database, when we have a collaborator here from Universidad de Costa Rica, Professor William Zamora, and we have a participate also in the Colombian Natural Products Construction Database. So once we have it, what we are doing right now is start to playing with the data sets. So we have here we have a first paper when we use the entire data set of the Peru Natural Products Database, in which we study when we were searching for a compound against Leishmania infantum, which is a parasite that causes visceral leishmaniasis, which is a disease with high rate of mortality. It's about 90% of mortality with left untreated. So we saw that we selected three compounds, natural compounds, one from Mexico, one from Brazil, and one from Peru. And then we submitted it to molecular dynamic simulations in a very different way, which is but lowering the pH of the simulation, so we can have an idea of what is the behavior of the interaction between a target and a compound in a way that it's more nearly than it occurs in the infection, because when we have some pathogens that develops under a lower pH. So we have a couple of them passes through all the filters, and we are having right now planning in vitro and in vivo validation of these two compounds. So with the complete data set, we have worked with SARS-CoV-2, which is in the times of COVID, everybody worked with SARS-CoV-2. And then we have a recent paper working with the mutations of the MPRO from SARS-CoV-2, and we have a good results for one natural product for all the mutations. And then we have we got a paper on Alzheimer's disease with a polypharmacological approach, when we saw that one natural product can bind to several targets, which is a thing we are doing right now. I didn't bring the data, but we have a paper published in the beginning of the year where we select when we select a pathway from Leishmania and when I run an analysis for a multi-target, because necessarily when we run this type of analysis, normally we got one only protein, So, but proteins are a network of they just interact with each other. So, in this case, this type of parasite, they are very complex and they have mechanisms of compensation. If you block one enzyme, they may have some mechanisms that bring, the pathway onto a normal function. So it's this type of approach to have more than one enzyme blocked by a single molecule can be a way to have more efficient drugs for this type of disease. Well, which is upcoming. We have a work on Chagas disease that we are running. We have analyzed the entire proteome of the Trypanosoma cruzi and selected only two targets that have the quality of being present at the most important and most numerous pathways of the parasite, and that they are central for the network, and they are also different or absent from mammalian, which is important. And we are running the virtual screening assay against the LANA PDB, which has 13,000 of molecules, of single molecules. We have a LANA PDB is a initiative from Professor Jose Luis Medina from Mexico, and it comprises compounds from Brazil, Costa Rica, El Salvador, Mexico, Panama, and Peru. So it's a very broad data set. And finally we have a hepatocellular carcinoma type study that we are developing right now. We are not truly we are writing the paper when we employed that single cell data for differential expression on diseased liver and healthy liver, and then we run an analysis trying to select genes that were expressed only in cancer, in hepatocellular cancer, differentiating it from other type of cancer. So we got one molecule that run other filters for the virtual screening, and we are now having some molecular dynamics to to have them more accurately selected. So we are what are we doing is also we are we're working with another parasites like Toxoplasma gondii, antibiotics, antifungals, antivirals, other type of cancers. We have we are working on some neurodegenerative diseases. This is our work. Is not it's not also being developing with emerging contaminants like microplastics, and a quite new thing is therapeutic molecules, which we are seeking to design with natural products as devices, nano devices to deliver drugs. So that's a new thing we are doing. I hope the next time we are going to have some results. So I want to thank and present my team, a young team. We have only 2 years of functioning with the research group. So I do appreciate your attention.
Thank you, Miguel. Excellent presentation. Any questions? Yes, we have a question here.
Thank you for all the good new ideas that you're bringing into the group. I wanted to ask you if you can say on the Trypanosoma cruzi, or cruzi as they prefer in English, have you looked into, I mean, I wanted to ask you what your targets were. I mean, obviously, I don't know if you can say or not, but crucipain or something that has to do with the acidocalcisomes, probably, no? Oh, so we'll have to talk in private. Okay, well, thank you very much.
Any other question? Yep. So we go to the next. Now we will have with us Professor Ryan Nett, who is going to present on understanding and harnessing the diversity of plant chemistry and metabolism. Dr. Nett is an assistant professor of molecular and cellular biology at Harvard University. His work focuses on understanding the chemistry plants use to synthesize diverse bioactive molecules important to human health, drug development, and agriculture. Welcome, Professor Nett.
It's a little higher. Does this sound good? Yeah. Okay.
Let's try with.
Okay. Does this sound good right now? Okay, great. I'll get started then. Make sure I actually know how to work this. Okay, so this is also my first time participating in this meeting and my first time to Costa Rica. So this has been a really nice experience so far. It's been great meeting all of you. I hope I get a chance to talk with you all in a little bit more detail. And yeah, I'll tell you a little bit about the work that my new lab has been focused on, what I've been focused on in the past, where we're really trying to understand how plants build molecules of interest. We could potentially harness that chemistry and use it for our own purposes. Just click. Okay. There we go. Okay, sorry about that. So I'm not going to belabor the point. I think many of us, if not all of us here, are very much invested in the importance of plants as medicines, and we're impressed by the diversity of plants that are used as medicines, as well as the diversity of molecules that are produced by these plants. And this is what we're particularly interested in my lab, are these different structures that plants can achieve and produce that can hit different molecular targets in our bodies and cause a number of different effects that can be beneficial. And so I'm showing you here some famous examples of molecules that, in this case, are FDA approved for use in the United States, but have also been used historically as medicines in different contexts. However, one of the major challenges with using these molecules, or really plant natural products more broadly, is that accessing them at scale can often be a challenge. And this is for a number of different reasons that most of us are familiar with. The plants sometimes are not easy to cultivate. They could be slow growing. They could be endangered. And the molecules themselves might not accumulate to very high levels. And so we have this inherent challenge of actually accessing these molecules. And so what our vision is in my group, and there's others working on this as well, is that if we can understand how the plants actually build these molecules, we can then take the genes from these plants and put them into other organisms and scale up production. And the reason that we're able to do this, or we think we can do this, is that ultimately all of these specialized natural products that are present in plants that have interesting functions are derived from the same primary metabolites, amino acids, isoprenoids, and acyl precursors that are present in essentially all organisms, or at least in all plants. And so if we can identify these enzyme catalysts that can take these precursors and convert them into these specialized natural products, we can then think of interesting things to do with these genes and enzymes. So for example, we can take pathways and put them into something like yeast and try to scale up production of our molecule in a bioreactor. Or we could even think about taking a plant metabolic pathway and putting it into another plant that's easier to grow. Perhaps we could grow it in a greenhouse or in a field. And I'm not going to talk about this in a lot of detail, but something else that I'm very interested in is that because most of these plant biosynthetic pathways have not been characterized, they also present a really great opportunity to identify both new enzyme classes as well as new catalytic reactions that have not yet been described in nature. But what I want to focus on today and tell you about is the process that we actually use for finding biosynthetic genes in plants. How do we actually find these enzymes and reconstitute these pathways? And the system that we've settled on, and something that's been really useful for our group and many others, is relying on the fact that the production of specialized metabolites in plants is typically conditional. So we don't have production of these molecules in all places at all times in the plant. It's usually under certain conditions. For example, in a particular tissue type, during a particular stage of development, or perhaps under an environmental stress. So you could imagine, for example, an insect chewing on a plant leaf and inducing the production of a compound of interest. And so correspondingly, if the molecule is only being made in certain places at certain times, we would then expect that the corresponding biosynthetic genes that are making these molecules are only going to be expressed where the molecule is actively being made. And so it's very simple in concept, but it's actually a quite powerful strategy for identifying biosynthetic genes. So what we can first do is perform a metabolomic analysis to try to determine an active site of biosynthesis. And there's different ways that we can do this, isotope tracer studies and just using LCMS to profile where the molecules are accumulating. We could look at different tissues, for example, in the plant, and try to identify an active site of biosynthesis, and then use this information to guide transcriptomic profiling of our plant to try to associate gene expression with biosynthesis. And this is relatively cheap and easy to do these days using RNA-seq, where we can sample different tissues, measure gene expression, and try to build co-expressed networks of genes that are likely involved in the biosynthetic pathway of interest. And so this is our general strategy that we use within my lab. I'll talk about it in a little more detail. But broadly speaking, within my group, we're working on many different compounds. Many of them happen to be alkaloids. I'm showing some of them to you here. So for example, we work on this molecule, huperzine A, which has been explored as a treatment for dementia. In the past, I've worked on colchicine, which is an anti-inflammatory drug used for treating gout. And we're also working on some molecules that have some potential as use as antidepressants, including this molecule harmine, a harmala alkaloid, as well as this molecule mesembrine. And to sort of highlight the process that we use for biosynthetic pathway discovery, I want to talk to you about this molecule, huperzine A, which we've been working on for some time now. So huperzine A, or I'll refer to it as hupe throughout this talk, is a major alkaloid that's produced in club mosses. So these are early diverging land plants that belong to the Lycopodiaceae family. And the reason that huperzine A has generated a lot of interest is because it acts as an acetylcholine esterase inhibitor. So it's able to reversibly and potently inhibit this enzyme acetylcholine esterase. And this has led to general interest in its use as a treatment for different neurological diseases, in particular as a potential treatment for Alzheimer's disease. More broadly, though, huperzine A is just one out of hundreds of lycopodium alkaloids that are produced in these plants. And so to date, there's been 500 described unique structures. They have many different chemical scaffolds with different oxidative tailoring decorations that can occur. And before we started working on this, the biosynthesis of these molecules was largely unknown. We really didn't know anything about how these molecules were actually made in nature. And so for me, as a sort of person very interested in plant chemistry and basic science, this seemed to represent a really good system for finding new enzymes and new chemistry. On a more practical level, huperzine A suffers from many of the challenges that I've described earlier, particularly challenges in sourcing. In particular, hupe is currently sourced from the native Huperzia plants that grow in Southeast Asia, and this has been very damaging to those local ecosystems and is not particularly sustainable for obtaining sufficient quantities of this molecule. So if we can understand biosynthesis, we could potentially produce it in other systems. And so if we want to understand how hupe is actually made in these plants, the first thing that we want to do is obtain a hupe-producing plant. In our case, we worked with this species Flammariurus tetrasticus, and we try to figure out where this molecule is actually made within the plant. And I don't have time to go into all the details today. We did a number of different experiments, including isotope tracer studies. But essentially what we were able to show is that this new growth tissue at the very tip of the shoot seems to be the place where huperzine A accumulates to the highest levels. Although you can find it elsewhere in the plant, this seems to be the active site of biosynthesis. And so with this information in mind, we can then move forward with RNA sequencing of our plant tissues, both biosynthetically active tissues as well as inactive tissues, so we can do differential expression analysis, and then try to find which transcripts are actually involved in our biosynthetic pathway. However, as you might expect, this becomes a bioinformatics challenge as you have tens of thousands of transcripts that are present within the genome of these plants. So it becomes a little bit of a bioinformatics challenge of how do you actually identify the relevant pathway genes. And the way that we do this is by leveraging chemical knowledge that we have of the pathway, or at least hypothesized chemical knowledge that we have of the pathway. We refer to this as biosynthetic logic. And we were fortunate that phytochemists had performed a number of isotope labeling studies in club mosses to define precursors to huperzine A. And in particular, these phytochemists were able to show that huperzine A is ultimately derived from an amino acid, lysine, as well as a fatty acid building block, malonyl-CoA. These isotope tracer studies further laid out that lysine is likely decarboxylated to form a polyamine, and that this is oxidized to form this heterocycle that I'm showing you here in blue. And they further show that malonyl-CoA is likely elongated to form this polyketide that is then condensed with this heterocyclic amine to produce these two precursors to the lycopodium alkaloids in huperzine A. And this was pretty much all that was known from these isotope tracer studies, but it did give us a starting place. And you could sort of rationalize from here how you could take these two molecules and put them together to form this more complicated scaffold that can then be oxidatively tailored to produce huperzine A itself. So on the surface, this is a lot of chemistry, and it might not be clear exactly where to begin, but we can actually use this information to our advantage to try to identify the relevant biosynthetic enzymes in these plants. And so in particular, if we focus on the beginning part of this pathway, we see that lysine is predicted to be decarboxylated to form polyamine. Well, we know from the literature that there are enzymes that can do this. Not surprisingly, they're called lysine decarboxylases, and these are very easy to predict out of a transcriptome. And so what we can do is look in our transcriptome specifically for a lysine decarboxylase that's highly expressed at the active site of biosynthesis, and then use this as a bait gene in co-expression analysis to try to find other genes that are likely involved in the pathway. And so in this pathway, what we might look for based upon this biosynthetic proposal, for example, are copperamine oxidases, which are known from the literature to catalyze this type of oxidative chemistry. We might look for polyketide synthases that can use malonyl-CoA as a substrate. And for these downstream oxidative tailoring reactions, we can look for enzymes like cytochromes P450 or 2-oxyglutarate-dependent dioxygenases, the major oxidative enzymes that plants use to carry out this chemistry. And so if we use this bait gene strategy, if we look specifically for a lysine decarboxylase gene that's highly expressed in the new growth of our plant, and then correlate the expression of all genes to this lysine decarboxylase, what we find is that out of over 40,000 unique transcripts in our plant, within the top 25 correlating to this lysine decarboxylase, we can find copperamine oxidases, type 3 polyketide synthases, and a bunch of different 2-oxyglutarate-dependent dioxygenases, or 2-OGDs. So the exact types of enzymes that we predicted to be involved in this pathway all fall out in this top list of candidates that correlate to lysine decarboxylase. And so it seemed like this correlation strategy worked pretty well, but these are simply candidates. We now need to actually test them for function. And so the way that we do this is using a system that I'll refer to as agrobacterium-mediated transient expression. I'm not sure if you all are familiar with it, but I'm going to go into some detail just so that you understand exactly what our workflow looks like. And so I explained to you how we identify our candidate genes. We can then clone these candidate genes as you would in any other system, and then put them into agrobacterium, which is a plant pathogen. We can create a strain library wherein each individual strain harbors a single candidate gene of interest. And as I mentioned, agrobacterium is a plant pathogen, and we can actually use this to infect and transform a plant host, in this case, a relative of tobacco, Nicotiana benthamiana, and actually have the plant then express our genes of interest for characterization. And so what this looks like is if we have a single agrobacterium strain with a single gene construct of interest, we can infect our leaf. We get transfer of our gene construct into the leaf, and then the plant machinery takes over such that we get gene expression and then production of our protein of interest. In our case, we'd be interested in enzymes, but you can do this, for example, with green fluorescent protein and see very nicely that you get this nice fluorescence after infecting the plant with agrobacterium. The really nice thing about this system is that we're not limited to one strain and one gene at a time. We can actually mix and match many strains simultaneously so that we can get co-transformation of many different gene constructs and the production of many different proteins at the same time within plant cells. This is important because this allows us to do combinatorial batch testing. So we're not just testing one enzyme at a time, but we can actually test them in groups of 10 or even 20. And this also allows us to build pathways in planta. So for example, if we identify a candidate gene that can convert molecule A into molecule B, we can then simply add in another candidate gene, candidate 2, that can take molecule B and convert it into molecule C. And so what we're actually doing here is producing a substrate in planta that otherwise might be inaccessible through chemical synthesis or isolation from a native plant. So we're able to build our pathway step by step as we identify new biosynthetic reactions. If we don't have access to substrates within the tobacco plant, we can also add in substrates exogenously, just as you would do to an in vitro enzyme reaction. And then after several days of incubation, we can extract metabolites and then perform either targeted or untargeted metabolomics to try to identify either substrates that are being consumed or new products that are made by our enzymes of interest that we're testing. And so bringing it back to huperzine and the candidate genes that we had identified, we were able to show that this lysine decarboxylase and this copperamine oxidase act together as expected to convert lysine into this heterocyclic imine that I'm showing you here. And we were also able to show that this type 3 polyketide synthase we had identified performs not just this chain elongation, but also the subsequent condensation with this heterocyclic imine to produce these 2 key precursors. You can see here from an LCMS trace, we only get production of this molecule when we have all 3 enzymes co-expressed at the same time. Simultaneously, we wanted to try to jump into downstream huperzine biosynthesis. And what I'm showing you here are a number of different lycopodium alkaloids that have all been isolated from huperzine producing species. And if you lay them out left to right, as I'm showing you here, you can sort of imagine how this could be a plausible biosynthetic pathway going from this molecule phlebelidine and through a number of oxidative tailoring steps getting through to huperzine. We were fortunate enough to have a collaborator from the University of Malaya, Yunyi Lo, who had isolated many of these alkaloids at milligram scale and provided these alkaloids to us to use as substrates. And so, as I mentioned before, we could actually take this molecule phlebelidine, for example, and actually add it into our leaf and then see if the enzymes we had identified could actually convert this into huperzine. And so we did this, and what you can see is if we have a GFP control and we add in phlebelidine, we don't get any consumption of our molecule. However, if we express a particular 2-OGD enzyme, which we're calling 2-OGD4, we get consumption of phlebelidine and the production of this new molecule that has this carbonyl. Similarly, if we add in a second 2-OGD enzyme, this 2-OGD5, we get consumption of this molecule and the production of this molecule that introduces this double bond. And we can do this stepwise, adding in new enzymes that we discover until we can convert phlebelidine all the way through to huperzine itself. And what I'm showing you here is a linear pathway going from phlebelidine to huperzine, but what we found that was really interesting is that we could actually mix and match these enzymes to access different alkaloids that are present in club mosses. And actually, if we add in different desaturases, we're able to achieve a lot of different chemical diversity that are found within these club mosses. In fact, we were able to access 16 unique alkaloids just by using these enzymes in different combinations. And I think one thing that's sort of an interesting observation that comes out of this is that if we look at the acetylcholine esterase inhibition activity of these different molecules, it seems like biosynthesis is proceeding towards a more potent acetylcholine esterase. And so, for example, if we look at huperzine, which has an IC50 of 82 nanomolar for inhibiting acetylcholinesterase. If we go back just one step in the pathway to huperzine C, we can see this is quite a bit higher with a IC50 of 0.6 micromolar and even higher with huperzine B. And if you go back one more step, you don't even have any inhibitory activity. And this is generally true with these other alkaloids that we can isolate, that have been identified from huperzine A producing plants. And so it seems like the plant has evolved to make a more and more potent acetylcholinesterase inhibitor. At least that's our speculation right now. And so just to summarize what I've talked to you about so far, we've identified these early steps in the pathway, which we can call an upstream module, and we've identified these downstream steps in the pathway, which we can refer to as the downstream module. And of course, we want to achieve full biosynthesis of huperzine A, and I don't want to belabor you with the details because a lot of it's the same and it gets sort of into the weeds chemically. But more recently, we've been able to identify the final steps in this pathway and connect the biosynthesis of this early module all the way through to this late module. So we now have enzymes for every single one of these biosynthetic transformations. And this was work that was done by a graduate student very recently in my lab, Eric Fields, who I helped to identify the final enzymes in this pathway. And essentially what he was able to do is take all of these 20 genes that we had identified, put them into the tobacco leaf, as I explained to you with agrobacterium, and show that we could indeed reconstitute biosynthesis of both huperzine C and huperzine A in the tobacco plants. We effectively moved a full biosynthetic pathway from club mosses into tobacco. And something that we're really excited about and something that's very new that we found is he's actually identified additional auxiliary proteins, or that's what we're calling them, that actually boost the level of both huperzine C and huperzine A through mechanisms that are not entirely clear to us right now. But this suggests that there's other proteins in addition to enzymes that are important for these pathways to move forward optimally in heterologous systems. And this could be important for engineering these pathways in the future. Okay, so this was our work with huperzine A, and I think this really nicely illustrates the process that we use for pathway discovery. And I really, quick before I finish up, I want to highlight some of the other work that we've been able to achieve with the same exact strategy. And so I'm not going to go into all the details, but just show that this strategy can be effective for many different plant natural products. And so first I'll start by talking about colchicine, which is a molecule that I worked on during my postdoc. This is a mitotic poison and has been used as a gout medicine. It's produced in the plant Gloriosa superba. And to spare you the details, we did things very similarly to what I described to you for huperzine A. We first performed a metabolomic analysis where we profiled different tissues in the plant to try to identify an active site of biosynthesis. As you can see here, we get most colchicine accumulating in the rhizome, suggesting that this is where it's being made. And we then paired this with transcriptomic analysis to try to identify co-expressed sets of genes that are likely involved in biosynthesis. And similar to the huperzine A story, we were able to identify a full biosynthetic pathway going from the amino acids tyrosine and phenylalanine all the way through to colchicine. We could take these 20 genes, put them into tobacco, as I explained to you before, and again, show that we can reconstitute colchicine biosynthesis within the tobacco plant. One thing that I want to point out, this is largely proof of concept right now. We're only able to produce maybe 1 microgram per gram of dry weight of colchicine, which is nowhere near what the native plant produces, but I think this gives us a jumping off point for trying to engineer these pathways in greater capacity in heterologous systems. The last example that I want to give you is for this molecule harmine, which is a harmala alkaloid. It's produced in many different plant species, but one of these that we've worked on is Peganum harmala or Syrian rue. And we know from previous work that this is ultimately derived from tryptophan. And so we wanted to understand where this molecule was actually being made in the plant. And so we performed a relatively simple labeling study. We fed in deuterated tryptophan to the plant and then looked to see where harmine accumulated the deuterated label. And what we found was that out of the entire plant, we really only saw label incorporation into the roots. So we have root-specific biosynthesis of harmine. And then we move forward with our transcriptomic analysis, much as I've already described to you. And very recently, this postdoc in my lab, Fabiola Muro Villanueva, has been able to find enzymes for basically every single step. Within this pathway, it's quite a bit shorter than the other ones that I've described to you. And there's still a few details to work out, but we can take the 6 genes that she's identified and put them into tobacco. And we're able to achieve production of this penultimate precursor harmaline. We still have a little bit of work to do to try to optimize biosynthesis to achieve production of harmine itself, but I think these results are really promising, and I think she'll be able to achieve production of harmine in the near future. Okay, so at this point, I want to wrap up and just leave you with a couple of major takeaways from the work that I presented today. If you remember nothing else, hopefully this is at least a nice summary. So I hope I've convinced you that we have a really powerful strategy now for accelerating the discovery of biosynthetic pathways in plants. We can actually go from medicinal plants and known molecule to full pathways in just a matter of months and years. Second, we can actually engineer these pathways in a model plant relatively rapidly. Part of this is due to the system that we use that allows us to test, discover, and engineer at the same time. And third, we've been able to use these biosynthetic pathways to actually achieve the production of new molecules. As I showed you with huperzine A biosynthesis, we can produce not just huperzine A, but many other alkaloids that are structurally similar, but might have distinct properties that may be useful in the future for different purposes. So with that, I'd like to thank you all for your attention. Obviously, I'd like to thank everybody that was involved with the work. I'll highlight again Eric Fields and Fabiola Villanueva from my lab who did the majority of the work. And if there's time, I'd be happy to take any questions you may have.
First of all, thank you for the presentation. All the plans that you mentioned that are part of your laboratory work these days are internal programs, or do you also actually partner with the industry in order to try to elucidate different pathways for talenting plant materials?
Yeah, we haven't yet collaborated with industry. This has mainly been looking at plants that we can easily access in our lab and obtain government funding for. So that's largely how we've been going about it. It's been very molecule-centric. We look for molecules that are interesting either pharmaceutically or structurally, and that's what we've been going after. But we're sort of open to many new directions. We're just sort of excited to discover new plant chemistry.
Yeah, do some of the co-expressed genes, do they co-localize in the genome? Do they form clusters? That's one question. Second, have you considered or tried CRISPR loss of function screens?
So for the first question, when we've looked for clustering of these genes together on the genome, it seems like there's some clustering sometimes. So for example, colchicine biosynthesis, where there's 20 biosynthetic genes, I think 2 or 3 of the genes cluster together on the genome. So not as promising as you would see in fungi or bacteria, for example. And it seems like this is broadly true in plant genomes with these specialized pathways. You get some clustering for some pathways, but in general, you don't get co-localization on the chromosome. And could you repeat your second question? We would really like to be able to manipulate the genes themselves in the native plant. Oh, I see. We haven't tried that, no. No, everything that we're doing is ex vivo. We're taking the genes and putting them in other systems, partly because of the medicinal plants being a little difficult to work with. Yeah.
So this is kind of a related question, but so the genes don't cluster, but have you looked at if there are any retrotransposable elements near them that tend to be expressed across different loci?
So we haven't looked specifically for that. What I'll say is that from looking sort of casually at the genomes, where these genes, like in club mosses, for example, for huperzine A biosynthesis, it seems kind of littered with retrotransposons around these genes. So I don't really know if I can't personally make heads or tails of that. I have noticed that some of those do co-express. If we look at our co-expression data sets, you do find those elements within the top candidates. And I've never really known if that's an artifact or if that's real, and that's not something that we've pursued, but it's interesting for sure.
Thank you, Ryan. Thank you. And now we are going to change the subject and we are moving into funding. And for that, we have invited Dr. Flora Katz. She is the director of the Division of International Training and Research at Oyarty International Center at the NIH. Dr. Katz is a global health leader at the Oyarty International Center as a director of the Training and Research Department. She oversees innovation, drug discovery, genetic, and informatic programs to build research capacity in low and middle income countries. Her work reached science, policy, and development. Welcome, Dr. Chavez.
Oh, thank you very much.
See you.
Are you going to drive these slides? It's a very small box for me to see, or should I share my screen?
Yeah, we are.
Excuse me?
You listen to me right now. Are you hearing me?
Yes, I am.
Great. Yeah, you need to share your screen.
Okay, I will do that.
Thank you.
Okay. Okay, well, thank you very much for inviting me. It's really nice to see some of the names and faces of colleagues I've worked with over the years in the ICBG. So hello to all of you. I'm going to talk briefly about funding opportunities for natural products research from the Fogarty International Center. And I'll just start by mentioning the Fogarty International Center is one of the 27 centers and institutes at NIH. It's the smallest one, and it's the only one that's really focused exclusively on international work. Our mission is to address global health challenges, and we do that by focusing on building research capacity in low and middle income countries globally. So all of the opportunities I'm going to talk about are meant for low and middle income country investigators or collaborative research with US institutions. We currently have 25 active programs, and you can see most of our work is in training, educational development, and career development. We only have a smaller slice that's on research, but I think that opportunities will be of interest. So I've selected a few things to mention that I think will apply to the natural products field. We have nothing that's specifically targeted for that area. Very important resource is our website. If you Google Fogarty International Center, this will come up. Has a very nice search function. You can see it has a number of tabs, particularly the program tabs, which explain all the programs one by one, and other tabs that are relevant. So I'll refer back to this later. The first opportunity I wanted to mention are our Career Development Awards. These are 3 to 5 year fellowships for mentored global health research and career development. And we have 2 versions. One is the KO1, which is for advanced postdoctoral investigators and early junior faculty in the United States. And the other one is, and there are also K awards from all of the other institutes at NIH that one could apply for to do natural products research. We have a unique opportunity, the K43, called the Emerging Global Leader Award. It's specifically for junior faculty in LMIC institutions, and I'll give you a little more information on that. So if you go to our website, you can get a lot more information. If you Google Emerging Global Leader Award, Fogarty International Center, this will come up. You'll have the 2 solicitations, the deadlines, the eligibility, program overviews, and then a list of recent awards that link to the public database reporter that'll give you the abstracts and contact information for all of the previous awards. And very importantly, all of our web pages have a contact for a program officer. So if you're considering applying to anything, it's always useful to talk to the program officer and say, this is my idea. Is that going to be responsive? So for the K43 or the Emerging Global Leaders Award, you must be an LMIC citizen. It provides 3 to 5 years of mentored research and career development in any area of global health, in any area that NIH supports. So that includes natural products. a minimum of 75% effort for the award, and in this case, up to $75,000 salary and 30% research costs. You can't have independent funding already at the R01 level, but we hope you will transition to that during the 5 years of the award. You have to have both US and LMIC mentors, the majority of your time spent in the home country, so this is not meant for you to travel to a US lab and work there for 5 years. And the scope is broad, and we have many partners that are paying for these awards and very interested in them. And the K01 award is very similar, except you should be a US citizen or a non-citizen national or permanent resident. Everything else is pretty much the same. Except we allow advanced postdoctoral appointments as well as recent appointments as junior faculty. Okay. Now, the previous awards, you can see there's a lot of K01s or the US awards working throughout Latin America, but the K43 awards that are direct to the foreign institutions are largely in Africa because that's who applies. We do have some scattered in the rest of the world, including a few in Latin America, and we'd love to have more. Okay, the second category is research training. These are institutional training awards. Again, they're divided into different programs, but they pretty much cover every area of research. So we have a global infectious diseases training program, an HIV research training program, and a non-communicable diseases and disorders training program. So if the area that you want to train in falls into one of these categories, then you can set up a 5-year training program. It's about $250,000 to $350,000 per year. We're non-prescriptive about the form of that program. And the common features are that they have to provide training for LMIC scientists. This is not to train US students, and help build institutional capacity. It can be in any format, so it can be degree or non-degree, short or long, etc. It's usually a collaboration between an LMIC and a US university, but the US partner is not required. You can do some training in the US, but the research project must be located in the home LMIC country. And it may require an additional active research award as a basis for training. They're variable, so you have to read the funding opportunity announcement. And again, we have co-funding from many ICs, so we cover all areas. Given some of the talks today, I thought this might be a relevant research opportunity. It's called the Brain Disorders Across the Lifespan. It covers brain, mental health, and all neurological disorders at every stage of life. And it comes in 2 forms, an R01, which is the standard NIH grant for 3 to 5 years, and an R21, which is a smaller exploratory 2-year award. That doesn't require preliminary data, and you can use to produce the data that you would need to apply for a larger R01. These are all collaborative awards. Well, they're largely collaborative awards between US and LMIC investigators, and the research must take place in the LMIC. There's a deadline coming up this late fall in November and for AIDS in December. And we have 9 partners from other ICs. I think of most interest to you, the National Center for Complementary and Integrative Health, NCCIH, just joined this program, and they're interested in supporting basic, mechanistic, and translational research on natural products and their therapeutic potential to treat nervous system disorders, in particular with relevance to pain management, but not exclusively. There is a notice that they put out announcing that interest and the linkage to this program. And if, again, you Google Fogarty International Center, brain disorders, that webpage will come up, and the link to this notice will also come up. I put in a couple of more opportunities just in case it applies to anybody. This is from the National Institute of Allergy and Infectious Diseases, NIAID, and they have a RFA out, or a solicitation out, specifically to support applications for high priority, regionally relevant infectious disease research in resource-constrained countries. And they're most interested in LMIC applicants, and only foreign applicants are eligible. So it's a constricted eligibility pool. Most of these are collaborative projects, but the US partner is not required. Any aspect of infectious disease research, so natural products approach to that would be completely responsive. They're small R01, so they're smaller budgets, but you can get 5 years of support. And if you think you're interested, definitely call the program officer that's listed on their webpage to see if your proposal would be responsive. You just missed the August deadline this year, so the next deadline is next August. I've also listed 2 other of our research programs just in case. If you're working at the intersection of HIV and non-communicable diseases, we have an R21 program. And if you're doing anything in mobile health, we have a R21, R33 program. Finally, just as a tool, if you go back to our website, Fogarty International Center, there's a funding tab. And under that, we post funding opportunities from the rest of NIH and also from non-NIH funders that happen to come across our desk. So this is not a comprehensive list of funding opportunities, but what we get sent a lot of material, and it may be useful for you to look at that occasionally. The other way to find out about these opportunities is to subscribe to one of our numerous subscriptions. We have social media pages, YouTube pages, LinkedIn pages. We put out a newsletter monthly, and we have a weekly newsletter that goes out electronically. So if you want to keep in touch with opportunities for international research and training, this is a good source to do it. Okay, I'm stopping early because I thought there may be questions. So I'll stop sharing. Yes.
Do you have questions? Thank you, Dr. Katz. Any questions? Yes.
Yes.
Huh? From Panama.
Hello, dear Flora. It's so good. I wanted to ask you if for the considerations of granting these, countries like Panama would still be considered LMIC, or we are not longer eligible for funding?
As far as I know, you're still eligible.
Really?
Yeah, you can. I will check the World Bank category, because unless it very recently changed, you were eligible. So I can send you that. I can confirm that.
Thank you so much, Flora. Have a great day.
Thank you. Nice to hear you.
Any other question? Okay, so thank you very much, Flora, and we will write to you with more questions. And I think I'm happy about your answer. Yeah? And now, thank you. We change to a new subchapter. We are going to talk about large bank of extracts and local capacities. Yeah? The first presentation in this section will be from Dr. Marcelino Gutierrez from INDICASAT, the Center for Biodiversity and Drug Discovery in Panama. He had been a member of INDICASAT scientific staff since 2008. He has established a research group that works on bioprospecting using microorganisms associated with plants and marine organisms. And his group has produced more than 40 scientific publications on this topic. Welcome, Marcelino.
Thank you. Thank you, Veronica. Well, I'm going to share some of the projects that we are doing in Panama. So my talk is divided in 5 small parts. Like first, I will briefly talk about the Panamanian biodiversity, and then the symbiosis and the systems that we are studying in Panama. And then one example, coral-associated bacteria as producer of natural products. And another example, crab-associated endophytic fungi. And finally, some example with frogs. Okay, so Panama is considered a hotspot of biological diversity. In this map, we can see in black, the areas that have more number of species of vascular plants. So the areas in black have more species per area. So Panama is among the more megadiverse zones of biodiversity. So it's a hotspot of terrestrial biodiversity. Okay, and in marine environment, we can see something similar. So the circles in black are zones with a high level of endemism. And the circles in red, in white, are have big endemism, high level of endemisms, and also a high level of species richness. So you see here, Panama and whole Central America is a biodiversity hotspot for marine species. Okay, all this richness of species is joined by chemical diversity that we are trying to study. Okay, we have places in Panama like Coiba National Park is a marine protected area, and it was designated by UNESCO like a World Heritage Site. And in part, we achieved this because the scientific research that we're doing in this area. So Coiba has a high level of biological diversity and a high level of endemism, and is in the Pacific of Panama. Okay, to talk about a little bit about symbiosis. So we see this kind of examples in the nature when we see 2 compounds that are very similar, but they are produced by very distinct organisms that are taxonomically very different, like a sponge that produces onomide A and an insect that produces pederine A. So onomide A is a cytotoxic compound, and pederine is a defensive compound that produces huge pain to the human that have an encounter with this insect. So when people saw things like this, they start speculating about the real biosynthetic origin of these compounds. So they started thinking that very likely are not the macroorganisms, but the symbiotic microorganisms that live in association with these animals. Okay. Later in 2014, the group of John Piel in the University of Bonn in Germany found that the real producer of onomide 8 are a group of symbiotic bacteria in the genus Entotionella. These bacteria cannot be cultivated, so they made all this work using metagenomics. And actually, Entotionella is very likely the real producer of all the metabolites that were isolated from the sponge. Okay, in last year, they published this review where they published the proposed biosynthetic pathway for pederine and onamide A, confirming the real origin of these metabolites that are microbials and not from the macroorganisms. Okay, we have another example. This is a compound. This is a Jondelis that it was first isolated from a marine tunicate, Tennessee turbinata, and this compound is in the market for soft tissue sarcoma and also for ovarian cancer. So this compound was later found that a group of different bacteria produce very similar compounds. And actually, the group of David Sherman in the University of Michigan found that the real producer is this bacteria, is a symbiotic bacteria, Candidatus endoctrinatidia frumentensis. So it was demonstrated using metagenomics again that the real producer is a bacteria, is microbials. And same thing with the famous compound Taxol is actually produced by endophilic fungi that live in association with the plant. Okay, in this table, we see several FDA-approved compounds in different therapeutic areas, mainly in cancer, that were first isolated from macroorganisms, marine macroorganisms, and then they were found in microbes. So there is a trend to find that these drugs are actually produced by symbiotic microorganisms. So given this trend, we started to study several symbiotic systems that we have available in Panama. So one, the octocorals and fungus-growing ants, frogs, and later coffee-associated endophilic fungi. Okay, one of, for example, we isolated a family, a group of bacteria in the genus Pseudoteromonas, and we have these three here, and many of these strains are new strains. And we found, we have in this part the non-pigmented strains, and in this part the pigmented strains. And we can see that pigmented strains have a very potent antimicrobial profile. So we have here the targets, different bacteria and fungi, and we see here the color. So the darker the color, the more active. And we can see species of this one here that kill all the bacteria and all the fungi. So it is very, very potent antimicrobial. Okay, so we make an extract. We isolated this OT59 from this octocoral, and we see that it was producing inhibition of this fungi that was isolated from an eel coral. So there was kind of a rare phenotype here. So in the presence of continuous light, there is no inhibition. In the daylight cycle, there is a little bit of inhibition, and in the dark, there is a complete inhibition of the fungi. So what's going on here? So we make some experiment, and we use multi-tough imaging analysis. So using this technique, you can scan the inhibition area, and you can record all the mass spec, and you can give different colors to different mass specs. And then you can know the distribution in the space of the compounds. Okay, using this multi-imaging, we have here the same experiment. We have the fungi, we have the bacteria, and in the dark, we see that there is inhibition and there is the production of compounds here. In the dark light cycles, there is a little bit of inhibition, and you can see less compound, and in the light, there is absence of the compounds. Using this, as this is the mass spec that we're treating here. So with the replicated, these compounds, And we found that they were alteramide A and B. Okay, here we have alteramide A and B, and alteramide in this form, they are active, they have antifungal activity. And this rearrangement under the like is not active. So that was why this phenotype was happening. So compound is active in the dark and it's not active in the light because it's transformed. And we make a hypothesis that very likely the coral is using this compound to protect the coral when the polyps are feeding, so they get out in the night and they are more exposed to the attack of fungi, and that's why we have this the production of this kind of compounds. Okay, we have another example with coral associated bacteria, and we found this extract that have antimicrobial activity, and we make a molecular networking analysis, and we found this family of bromoalterochromides. Actually, it's important to say that we isolated, we identified this compound without isolating the compound. So we do it in the extract. Okay, so we annotated the compounds with the MSMS experiment, and we found 2 new compounds in this extract. And so this bromoalterochromide have antimicrobial activity, and it was published in 2020. Okay, another example, crop-associated endophilic fungi. You know that coffee, like all the plants, harbor a community of endophilic fungi. And this is, you see here, this is a fungi that is attacking the coffee. So the coffee have a beneficial fungi that protects against the attack of pathogens. And this protection is through the production of secondary metabolites. You see here, we're looking for this kind of inhibition halos. And when we find things like this, then we go to study the chemistry. So these fungi have an ecological role. It's protecting the coffee plant from the pathogens. Okay, we make an extract and do the biazo-guided isolation. We found that this compound is Menisporopsin A, has a broad antifungal activity and inhibit all these plant pathogens. We can see here that the activity of Menisporopsin A is very similar to the activity of mancozeb. That is a agrochemical that is used to control fungal infections. So in some cases, it's very, very similar activity. Okay. And also, we study these, isolated all these metabolites from crop fungi associated to coffee and to cocoa. And we found that they have activity in different assays. So we found 3 compounds active against Chagas disease, 4 active against leishmaniasis, 4 active against malaria, 1 antibacterial, and 4 active against the coffee rust that is a coffee disease. Okay, so this means that this compound have an ecological role, but they also have applications in biomedicine. Okay, this is a different example. So we found these compounds not in bacteria, but in these secretions that we obtained from the frogs. So we isolated these bufadienolides that have antitrypanosomal activity, and there are new compounds also, and also from the oocytes of these same species, we isolated these new compounds that have activity against T. cruzi. Okay, as a conclusion, our results confirm that the biological diversity from Panama, in particular symbiotic microorganisms, beyond their intrinsic value, have a great capacity to produce molecules with potential in drug discovery. And this is the research groups. We have here grad students, technicians, and undergraduate students. Thank you. Have any question?
The virus or the, sorry. The fungus you're trying to target is the Roya fungus, right?
It's one of them.
One of them. Do you think eventually a treatment could be developed by using the actual endophilic fungus, as in like culturing it and spraying that?
Well, we are trying to find a biological control. So trying to control the Roya using the microbes. And we have several very good candidates. And also we are exploring the chemistry, to find the compound that is active. So I think we have very potent. We're starting to study. Questions?
I think more questions. Thank you very much, Marcelino. It has been great having you with us in person. And now we are going to move to the next presentation. Matt Smith. He's going to the basics of barcodes and their uses. He's a professor at the University of Florida Department of Plant Pathology and also the curator of fungi at the Florida Museum of Natural History. He teaches fungal biology and conducts research on fungal systematics, ecology, and evolution. He's an author of more than 175 publications on fungi and is a fellow of the American Association for the Advancement of Science. Welcome, Matt.
Hello. Thank you so much for inviting me to speak with you. I'm sorry that I'm not there in San Jose instead. I would love to be there with you, but instead, I'll present from here. So I'm going to share my screen. And maybe you can tell me if people can see it. Hopefully people can see it.
Yes, we do.
Okay, great. So yeah, today I think I'm a little bit different from many of the people in that I'm not really focused explicitly on producing natural compound or obtaining natural compounds. I'm really a fungal biologist, but I'm going to talk a little bit about the importance of DNA barcoding and also sort of how they're used and why I think they're important. And this is from the perspective of fungal biology, and so I'll give a little background. So as I said, our lab really works on fungi. I'm very convinced that fungi are cool. That's why it says that there. And so I'm going to talk about some of the challenges that we have in studying fungi. So first, we have very high species diversity. There are somewhere around 130,000 species described, but we know that there's at least 1 million species, but probably more like between 1.5 and 5 million species of fungi on the planet, based on a lot of molecular data that we have. And so we really know that there's a lot we don't know about fungi, and that makes them very challenging. And some other related things that are intrinsically linked with that, they have very simple morphology. They have simple bodies. So most of them are made up primarily of mycelium, of their hyphae. And because of the way they grow and the way they interact with other organisms, they're hidden much of the time. So for example, within wood, or very specific substrates like animal dung. Also, as Dr. Gutierrez discussed, as endophytes in plants, so there's a very high number of species that seem to, at some point in their life cycle, live inside of plants undetected. And then we also know that in the soil, there's a very high diversity of fungi. So for example, mycorrhizal fungi. And also in water, more and more we're finding there's very high diversity there as well. So there are all these different habitats, and much of the time fungi are very simple. They're not evident to us. So we have to have a bunch of different tools to be able to study them. And then the other thing is, because their morphology is relatively simple compared to some other organisms, species are really tough to tell apart from each other. And this is part of where DNA barcoding comes in. So this is just an example from my own work in Southern South America. The genus Cortinarius, we know, is this hyperdiverse genus, at least 5,000 described species, and we know there are a lot of undescribed species as well. So getting a handle on diversity and trying to understand what are the different chemical features of all these different fungi is a real challenge for us. So while plants have maybe 300,000 described species, we're in the millions. So we're still in the age of discovery for fungi. And so that's important because fungi, we know, have already provided a number of really different kinds of natural compounds that are of pharmaceutical use. So especially antibiotics and antifungals, there have been a number of different classes. The first statins were all from fungi. There are a number of psychoactive compounds, and then also perhaps less known to many people are the immunoinhibitors that have been provided by fungi. And certainly there's a lot of promise for anti-cancer compounds. So these are just some of the main classes, and I won't go into detail about the history of that because I think the audience here probably knows a lot about those. So, but my point is, we know that there's a lot of secondary compounds that are being produced. And so trying to harness that requires trying to get a handle on this very high biodiversity. And so I think we have to step back and say, okay, well, what is a species of a fungus in particular, but also for other organisms that are challenging to tell apart from one another? So what is a species and how do we recognize those? And so there are different ways that traditionally that people have recognized species. So originally we always used the morphological species concept. And for groups like birds and mammals, it's relatively straightforward. So in this image, you can see there are a bunch of different pictures of primates. And if I asked you to choose the two pictures that go into one species together, it would not be hard for you to do that. And you could come up with a number of species pretty easily. But the morphological species concept has some problems when you try to apply it to some other groups of organisms. And when people realized that it wasn't that simple, they started to come up with other ways to try to recognize species. And so the biological species concept recognized that organisms that are able to mate with one another are the same species. So Mendel's peas are a good example, or in Central America and Latin America, you see jaguars, and there are two different color morphs, but we know that those are all part of the same species. So that's a good concept, but it really is very challenging for groups that we can't work with in the lab. And so what has really taken over, especially for fungi, but a number of other groups, is phylogenetic species concepts, where species are recognized as monophyletic groups, and that those are shared, they have a shared unique genetic history. And so we can use molecular data to diagnose those monophyletic groups. So this has also increased our understanding of evolution, of course, because we can look at relationships between species and determine how they're related to one another. And it wouldn't be possible just with morphology. So this is an example from the genus Agaricus. You will recognize it because this species right here is the most commonly sold mushroom in the world, Agaricus bisporus. And so you can also use that to get other kinds of information, such as chemical information about species, if you have enough genetic data. And we've heard some examples of that today. So these are 3 different edible species here. And then this one down here, Agaricus xanthodermis, is one you do not want to consume because it will make you quite sick. So it can also be, phylogenetic methods can be useful for understanding not just evolution, but things like edibility. Okay, so how does that fit into the context of DNA barcodes? So a DNA barcode should be a small piece of DNA that we can use to diagnose species. It should be something that's easy to sequence from poor samples of different types, but you also need to have a large library of known species. And as I said, we're still in the age of discovery for fungi. So we're kind of still in the age where we're building the library, but this is the point, the idea anyways. And for those of you who are not familiar with this or haven't thought about it a lot, this would be a case where you have several different specimens, specimens 1 through 3 up here, and specimens 4 through 6 down here. And you can see they have this DNA pattern that's different between the two groups. So you see variation between the species, but within a species, they're conserved. And then you could find a location inside the genome where you could sequence just a short piece of DNA to differentiate between these two barcodes and identify species. So that's the idea behind DNA barcoding, and it's becoming much more common over time. So I'm going to show some examples having to do with work that's happened from my lab using DNA barcoding and how I think it's important for capacity building and also for incorporating fungi more and more into searches for natural products. So this is that same DNA barcoding sort of graphic there, but just to say that one of the other main points about this is that with DNA barcoding, we can use all different kinds of samples. So I've been talking about specimens like mushrooms, but also cultures of fungi. But then we want to be able to use DNA barcoding for other kinds of samples. So like spore traps that can look at which fungi are there just as spores, looking inside of substrates like water or dung, or looking at the fungi that are colonizing roots. Those are just some examples of how DNA barcoding can be used. And I think it's important to drive home. I chose some examples that I think show that species identity has consequences. This is a relatively straightforward example from my lab where we work a little bit on truffle fungi. So Tuber brumale and Tuber melanosporum, shown here side by side, are two black truffles. And you can see there's a consequence to the different species because Tuber brumale is about $400 US per kilogram, whereas Tuber melanosporum is a little over $1,000 per kilogram. And what we did was use DNA barcoding to look at those fungi. A student of mine received samples from all over the Eastern North American region where people are trying to grow Tuber melanosporum because it's the higher value truffle. And using basic DNA barcoding and some phylogenetic analysis, you can see that Tuber brumale has been accidentally introduced throughout North America. And there was very few of the samples that we received that were the Tuber melanosporum, which is what people were trying to grow. And some of these sequences came from truffles and others came from mycorrhizae samples. So roots that were colonized by fungi. So this is one application of DNA barcoding in action. Of course, this one is not for understanding chemical compounds that can be used for human health, but the idea is the same. Okay, another one that we have worked on in the past where species identity had consequences is this mushroom reishi or lingxi, which has been called the mushroom of immortality. It's been studied and included for a long time in part of Chinese medicine. And there have been a large number of identified compounds in this mushroom, which was always identified as Ganoderma lucidum. Well, more recently, it's been recognized there are multiple species. And so there have been antimicrobial, anti-cancer, anti-diabetic, and anti-inflammatory compounds recognized in these fungi, but often under the name Ganoderma lucidum. And these products are sold in the United States and also throughout other parts of the world. But one of the issues has been which mushrooms really are being sold and consumed. And so in this paper, which was published back in 2018, we did DNA barcoding of different reishi mushrooms from grow kits and also from consumables. So these are things people are eating all over the world. And we purchased the ones that were available in the United States, but these are available in many different countries. And what we found was that that there were 10 different species that were all being sold under this name, Ganoderma lucidum. And the big question there was whether all of these fungi really have the same medicinal properties or health impacts. And also going back in the literature, the things that have been coming from Ganoderma lucidum, we don't really know which one of these groups they're coming from. And so this is a good example that has potentially big impacts for human health if people are consuming fungi that make chemical compounds, and they're not well characterized. So we don't really know what we're doing in a lot of cases. In other situations in this, they were actually growing fungi that were from other continents, and it looks like some of them have been introduced as well. So that's a separate issue, but another usage of DNA barcoding. And so I'm gonna give an ecological example because it did have something to do with capacity building. This is a project of my student, Marcos Cayafa, who's from Chile. And we were doing work in Chile, and we recognized some initial evidence that these two birds seem to be interacting a lot with fungi. So we saw situations like this below trees where these birds were feeding. And we saw that some of the things in this picture are truffle-like fungi. So the ones not in the circles are truffle-like fungi. And the other ones are fruits there. And we also noticed there were a lot of fungi with peck marks in them. So it looked like birds were eating them. And we found fungi that looked a lot like animals we were finding. So in this case, grubs and slugs and also seeds. So we wanted to ask this question, do birds consume truffles using this DNA barcoding approach? And to do that, as part of this larger project, we needed to build this library of fungi to compare to. So in that process, we barcoded about 2,500 fungi from all over Chile and Argentina in the region where we found Nothofagus trees and where these birds were present. And I think an important part to highlight here is that to do that, we really needed to have a very large team of researchers. And I think this is one of the most important things, at least for a mega diverse group like fungi, is that to really make progress on these organisms, we need to build capacity and build teams. So we had more than 15 researchers from 7 countries, and I'm showing you some of them here. And so I think actually we made a lot of progress in understanding these fungi and the interaction with birds, but perhaps the most important thing we did was educate among all of us, where we got a chance to teach each other. So I will go through it really quickly, but basically the short end of the short sort of synopsis is that we found that fungi were quite common using this barcoding approach based on next generation sequencing and sampling across this region of central Chile. We found that geography influenced the fungi that were found, and perhaps most interestingly to me, we found that many of the fungi were consumed by both species of birds, and that these fungi were abundant, especially the truffle-like fungi, which had no other way to get their spores out into the environment. And so this is just a picture showing you what some of these look like. And all of these were identified using a DNA barcoding approach so we could match the fungi from the library of collections to the sequences from inside the fecal material. And there's a nice publication on this by Marcos Cayafa and a number of co-authors, which I would invite you to read. So I guess the take-home message for me is that in working on a really, really diverse group like this, that barcoding is going to be really important for the future, and it's going to be really important for making connections between people who are working on the chemistry and the people who are finding these organisms in the field. And so sort of my prescription for the things I think that a lot of scientists, we need to do better. So we need to make science fun and provide resources for people. We need to build teams always to really be able to make progress. And I think we need to engage with young people to get them excited about our study groups. And the same is true for chemistry. And also interact with citizen scientists, because that's the only way we're going to be able to really work on these mega diverse groups to try to incorporate them into our studies on chemistry. And then lastly, I just wanted to thank you for your attention and for inviting me. I hope that it was something of interest, and I'm glad to take questions.
Really an interesting presentation, and we want to get in contact with Kaya, of course. Any questions? Yeah.
Really fun talk. So I have a question about DNA barcoding and nanopore sequencing. Is that something you see becoming more common? And also using adaptive sampling to limit the set of species that you can pull out?
Yeah, I think that there are a number of different approaches. All sorts of high throughput sequencing are being used. In my lab, we're using mostly Illumina and PacBio. But Nanopore, I think because people can do a lot of sequencing in the field, it's got some really great aspects to it. So I think people are going to be experimenting with all sorts of methods. And I think the interest in the chemistry of these fungi is growing. So I've heard, I was just at the International Mycology meeting in Europe last week. And there are a number of different groups all over the world that are using a lot of different innovative techniques. But Nanopore is just, I would say, just one of the approaches that people are using. I would say for DNA barcoding, a lot of the next generation sequencing technologies are great because you capture diversity within whatever it is that you're sampling. And if you have some sort of contamination or fungi living close together with one another, that you capture sequences of multiple organisms at a time. And so that's a real benefit. I don't know if that answered your question exactly, but hopefully so.
Any other question? Okay, we want to thank you, and we keep in touch. And
Thank
now,
you.
And now we will have our lunch, and we will convene back at 2:30 for our followers. Yeah. Thank you. Welcome back to the seminar Global Health, Biodiversity, and Therapeutic. This afternoon, we have a program with scientists from Latin America, but now the first one is a company, and very special company, yeah, and is led by Gaston Salinas, who is co-founder and CEO at Botanical Solutions. He is an industrial engineer that has passion built and around science-based ventures and corporate innovation. The knowledge and practice acquired in professional consulting has allowed him to translate a promising research project from university into an international business with growing revenues and a solid value proposition for multi-billion-dollar industries. Welcome, Gaston. You have the floor.
So thank you again, Veronica, for having me. I think this is the second time that I've been at this Congress, so thank you to the organizers. Just to set clear expectations, as Veronica said, I'm not a scientist. I'm not going to get into the nitty-gritty of science in this presentation. It's going to be more like an update on what we've been doing in the space of vaccine adjuvants at Botanical Solution for the last 18 months. But I wanted to kick off my presentation by sort of looking at this sort of phenomenon that killing an animal somehow may lead to different outcomes or considerations as opposed to killing a plant or a tree in this case. So I would like to spend a moment. Oh. All right. Is it better now?
I get it. If you want to look, we can give you the.
No, that's totally fine. No worries. Thanks. So I would like to spend a moment just to admire this tree. That is called the Quillaja saponaria. It's endemic to Chile, Peru, and Bolivia. It produces a compound called QS21 that has been commonly used as a vaccine adjuvant or coupled with protein-based vaccines. And it values, or I would say it's over 1,000 times more expensive than gold. Just to sort of give you a ballpark, a gram is about $400,000, between $100,000 and $400,000 a gram of this high value compound. But in order to get QS21, you need to literally kill a tree, a very old tree. And the reason for that is that saponins in general accumulates in the inner bark of the tree. So the older the tree, the more yield you get from this type of raw materials. So on this article, the Telegraph, they say that this is a tree that could help to cure malaria. As a matter of fact, QS21 is already in 2 blockbuster vaccines, Shingrix and RXV. The first one, I would say both of them are blockbuster vaccines. GSK reported sales by the end of last year in excess of $5 billion. So it's clear that GSK is going to need several kilograms of QS21 in order to keep on growing this business. These days, these 2 vaccines represents almost 50% of GSK's vaccine business overall. There is some hope, and GSK has made a public announcement that they want to become a deforestation-free company by 2030. So in that sense, we'll see what opportunities lay ahead after that announcement. But one of the reasons why a company like GSK is so tied to the use of tree bark as a donor material for making QS21 is basically regulations. And once you get an FDA-approved vaccine, is extremely difficult and costly to make any major changes to the downstream manufacturing system. So here you can see 4 simple images that somehow summarizes the way that QS21 is actually produced. So you start with trees, then you use the bark, you make crude extracts, you go deeper into purification until you get to the end-use product. So this is the second part where I'm going to be talking about updates on what we've been doing in the space of making a sustainable source of QS21, basically or based on a plant tissue culture platform that we originally designed for agriculture and that we have managed to get repurposed for the pharmaceutical space. So 4 simple pictures just to try to illustrate what we do in our lab. So we start with a small plantlets that are in average 30 to 60 days old that we grow in bioreactors. So we harvest biomass in a weekly basis. We make crude extracts and we're able to get to high purity compound. I would say that the 3 main pillars of what we do is basically starting with quality, because no matter if you compare 1 gram, a kilo, or a ton of our dry biomass, the chemical composition is almost identical batch to batch. When you rely on natural sources of raw materials, this is an important issue. Then, in terms of supply, we're able to make virtually unlimited quantities of those materials that allow us to serve the different business opportunities that we operate at the company. And from an environmental point of view, we don't harm any tree or whatsoever. Our approach was awarded the Best Process Innovation by an UK organization last year, and we're really proud of that recognition. So I started talking how difficult it is to make changes, especially when you have an FDA-approved vaccine. But I'm glad to share with you that we've been very successful in confirming that by changing all trees by our plant material, it's possible to be fully compliant with what is approved by FDA in terms of QS21 specification, and of course, from a production point of view, regarding scale and cost. This is not something that we have, I would say, validated only internally, but we have partnered with big pharma companies to validate the biochemical characteristics of our QS21 and the biological activity, but also other key intermediaries have been tested by big pharma and confirmed that all the parent pieces that we have designed in this production system could be or should be compliant with FDA-approved vaccines. Now, when we look into the space of, I would say, a sustainable QS21, we understand that we're not the only player. Other companies developing novel vaccines that may need QS21 in the future, they no longer need to rely on tree bark for getting their vaccines formulated. So these days, there are groups working on production of QS21 based on yeast, other groups using tobacco plants, and also other groups working on cell culture, Quilleja saponaria cell culture approaches. So the question is how our technology or our approach somehow compares to the other approaches of these other groups. And I would say what we have here is basically a table where we look at yield of QS21, current scale in terms of production of QS21, and last but not least, different purification approaches that different companies like ours have been following in order to make production of QS21 truly scalable. When you look at the first, I would say, row looking at QS21 yield, our plant materials in average contain between 0.25% and up to 1% QS21, just to give an idea how much is the yield of QS21 in all tree. Premium tree bark is about 1.5 to 2% QS21. whereas engineered yeast has been reported about 0.0012% of QS21. There is no public information about tobacco plants producing QS21, and cell culture could be somewhere comparable to what we're doing. What's the additional benefit of using our approach is that these days there are technologies that use enzymatic conversion in order to convert different saponins into QS21, and that could potentially allow us to increase the yield of QS21 up to 5%, depending on the lines that we put into production. Other systems that are focused specifically on QS21 cannot get benefit from that advantage. In terms of current scale, in our site in Chile, we can make as much as 5 kilograms of QS21 as a proxy of raw materials that we're growing in the lab. And coupling that with novel purification systems, we believe that our approach should be the most competitive, not only for FDA-approved vaccines, but also for new approaches for developing novel therapeutics. If I had to describe the current business opportunity, keeping in mind FDA-approved vaccines and new therapeutics, so basically I have outlined different target customers, competitors, and how we believe that we can compete into those spaces, starting with FDA-approved vaccines, where basically we're competing against Trivar. We believe that the sustainability factor and the IP that we have generated in order to grow the biomass and the purification system should give us an edge in order to compete into that space. Whereas on the side of developing novel vaccines containing with QS21, we believe that our current scale and purification approaches are the main in order to make us or allow us to compete into that space. The good thing about saponins is that these days there is an appetite not only for QS21, there is an appetite from the industry in other saponins, just like QS7, for example. And this is just a press release that was sent out by Sanofi a few weeks ago, where they invested about $1.5 billion, $1.2 billion in Novavax just to get access to a different vaccine adjuvant system that contains different fractions of saponins. And we believe that, again, we can couple our technology in order to be the most reliable and sustainable source for this type of compounds. As I said at the beginning, we started the company back in 2013 in agriculture, basically de-risking a technology to make, I would say, or to build a scalable platform for growing this Quillaja saponaria plant material. We have partnered with companies like Syngenta and more recently with Croda when it comes to get this repurposed for the pharmaceutical space. Last but not least, and I'm using sort of this event as a platform also to announce that we have already closed a Series A extension, which will allow us to bring or use that funding for production or in-house production of pharma-grade QS21, hopefully later this year. So this is one of the major milestones that we're planning to hit for 2024. So really excited to share this news with you today. Thank you.
Yeah.
What's the mechanism of the adjuvant effect of QS21? Is there more than one mechanism?
That's a good question. I think there are different formulations that you need in order to use QS21. Most of companies, or GSK in this case, develop AS01B as an adjuvant system for a specific immune response. I don't have the details behind what's behind AS01B, but yeah, I cannot comment much details on that.
And also, can you produce it? Do you know the biosynthetic pathways that lead to the production of QS21? Can you reproduce it in a heterologous system.
Yes, there is a group in England that have been trying to use tobacco plants for expressing QS21. In the past, have been tried a fully synthetic version of QS21. There were too many steps involved in the synthesis of the molecule that made it economically, inviable, or it was not feasible to make it. Last company trying to push that approach basically closed operations last year. And the other options were basically the ones that I outlined throughout my presentation.
This goes along with the previous question. So Ginkgo Bioworks recently got completely gutted. How would you avoid that happening to your company as well? Ginkgo Bioworks laid off like 400 people or something like that. And it's very similar, the approach, but I'm wondering how would you avoid being similar?
I would say I can share with you all the details, but I think we have gained significant traction with big pharma companies interested in our approach for making QS21. And as I said, it's not only for novel vaccines. We can use our approach and start selling tomorrow our compound, especially for FDA-approved vaccines. So, and I think that's probably our main competitive advantage today to other groups.
So I know at least for some plants, if you grow them in different conditions, in this case, a tree versus a sapling, it'll change the chemical composition fairly significantly. And that's been, I think, a frustration at times for people trying to do exactly what you're doing. I'm not sure if you can speak to this, but did you have to change conditions in order to get your plants to actually produce the 0.5 to 1% yields that you were getting? Or was it just sort of innate in the plants that you're growing that you can have them produce the right molecules at higher proportions than the other systems that currently exist?
That's a great question. Just to give you an idea, some of the elicitors that we use today for growing the plants allow us to boost yield of QS21 up to 4 times. And using, I would say, even lines that have very low yield of QS21. Of course, when you started working with higher yield lines of materials, you don't get gains as significant as the ones that you get with the lower yielding materials. But yes, we use different elicitors to elicitate the responses. We have seen that biochemical profile tend to vary compared to materials that you get from the wild. You need to avoid other peaks or other compounds that get closer to QS21 as well, because that in the end makes your life more difficult when you're trying to purify QS21. So yeah, we've been scratching our heads for a while, but good news is that, again, in the eyes of users or vaccine developers, our QS21 is as good as the QS21 or traditional QS21 that they're working with.
Thank you, Gaston. Any other questions? Okay, so then we move to our next presenter, Mr. Gustavo Carazo Berrocal, Director of the Industrial Department, Pharmacy Faculty of the University of Costa Rica. So he's one of our hosts. He is the coordinator of the phytochemical section LATIFEC. LATIFEC means Phytopharmacy, Pharmaceutical Technology Lab. Welcome, Gustavo, please.
That's the same. One more. One more look.
Hi, my name is Gustavo Carazo. I'm here from Costa Rica. I work here in the pharmacy faculty, and also I'm, like Veronica says, I'm the coordinator of the LAFITEC. It's a lab, a unit of the INIFAR, that is our research institute here of pharmacy. And I'm very glad to be here and to receive you in Costa Rica. I'm going to talk a little bit about what is the work that we're doing here in the INIFAR. We have like 4 or 5 years of working with some plants and looking for plants that have some medicinal characteristics. And we are exploring. We are beginning, but we think that we have to begin to get somewhere. Yeah, this is the first step. As we are talking about, Costa Rica is recognized for being one of the 20 countries with the greatest diversity of species in the world, and possibly will be the with the highest density of them. We have like 500,000 species in Costa Rica. That represents almost the 6% of the total world's biodiversity. Of which until 2018, more than 121,000 are known. We have a density of species like of 2.3 species per square kilometer. That is really high. The number of plant species in the country is estimated at more than 11,500. That approximately 12% of these are endemic, so that we can get it only here. The origin is from Costa Rica. Therefore, they are known only in Costa Rica, and it is not possible to find naturally in other countries. Some of like 500 species of these plants are, or we know, that have some medicinal characteristics. As you know, plants are studied really for medical purposes. In Costa Rica, we have this tradition of the use of these plants, mainly by orally from generation to generation. Also, there is another writing information that we can get, but we are losing some of this information. So in the INIFAR, we try to get some of this information to know how are they used in the population in Costa Rica. In some cases, the effects of plants in the treatment of certain diseases have resulted into an overestimation of the possible effects of these plants, and they attribute some properties that are not real. And this is what we are trying to look about. This is the INIFAR. This is the INIFAR, our Institute of Research. We have 5 units. First of all, La Yafa is the Pharmaceutical Analysis and Consulting Laboratory. Also, we have the LabioPhar, is our Biopharmacy and Pharmacokinetic Laboratory. Our youngest unit, that is the LAPONAVI, is the one is in charge of polymers, nanotechnology, and pharmaceutical biotechnology stuff. Also, we have the CIMET, is our National Center for Drug Information, and the LAFITEC, the one I work more specifically, that is the Phytopharmacology, Pharmaceutical and Cosmetic Technology Laboratory. So what we are doing, or example of what we're doing, we have an interdisciplinary team. We have biologists, we have microbiologists, we have pharmacists, chemists. So we are trying to look at these plants and what useful could be for medicinal purposes. First of all, we make a lot of tours with the biologists to look around Costa Rica for these plants. Then we make a botany workshop to have a plant identification of them. Then we have to collect the samples. And then we have to make extracts. We have the extractions of them. And we make some phytochemical tests to make a characterization, a phytochemical characterization of these extracts with different techniques. Then we have some tests, mostly in vitro and ex vivo, and some in vivo mainly with rats, depending what is the purpose of what is the useful of these plants that we are collecting. And then we have the results of the analysis. For example, this is a plant we collect in San Carlos. This is in the province of Alajuela. Then we have to make the extracts, and then we make some of the tests that we are trying to characterize phytochemically. First of all, what we are looking is for these medical plants, we make a research of the plants that were discovered between 2010 and 2020, looking for the possible usefulness for medical purposes. This is an example of some of these plants. We were looking for species that were from Costa Rica, endemic Costa Rica, that were recently discovered in these 10 years, 2010 and 2020, and that have ethnobotanical uses, or that the family to which they belong report such medical uses. With this research, we'll look some of these families. For example, I have two. This one, the Myrtaceae, the species that we look for them were the locations and some family usefulness that we can identify. This is also another example, the Passiflora, Passifloraceae, the species, also the locations, and the usefuls that we can, or we detect we can use. Another one is Polypodium, the Polypodiaceae. They're another example of these plants that we found here in Costa Rica. What are we doing now? In the 2023, we have 8 species that will be studied. Will be Mucuna pruriens and Mucuna urgens, Thinanthus coratinus and Thinanthus macrontus, Plinia costarricensis, Licania platypus, Witerigia solanacea, Ilocereus ondatus, Ilex aquilifolium, Gonolobus edulis, and Gonolobus palionarius. And in this year, we have 7 species that have been studied. Wigandia urens, Salvia polystachya, Banisteriopsis muricata, Molydenia viridiflora, Plinia costarricensis, the pitaya, is Zebra orejona, que es Purpusi ondatus, Thinanthus cratinus, and Passiflora that we are now studying. We make a phytochemical profile of first characterizations to know if it has alkaloids or it have terpenes, tannins, all of that. Only preliminary, only to have a characterization of those extracts that we have. And then we have to prove it. I have 2 examples of what we are doing. For example, this is the study of the effect on intestinal motility of fractions of the ethanolic extracts of Licania platypus in the leaves in an isolated organ model with rats, Sprague-Dawley, for identification of the extracts with the highest activity. We make the phytochemical results, carbohydrates, reducing sugars, proteins. Well, there is a lot of components. And we saw presence of tannins, for example, only in the extracts that were from Guanacaste and San Ramon. And then we look for possible pharmacology activities. For example, we suppose that we have an inhibition of the cholinergic pathway, especially to the non-competitive muscarinic antagonist effect on the release of NO by the cyclic GMP-NO system. And we suppose there is a possible mechanism on pharmacology action involved in the spasmodic effect of the metabolites present in these extracts of the leaves. The metabolites present in the ethanolic extract of leaves do not seem to interfere with the serotonergic pathway that participates in the contraction of intestinal mood muscles. And we look for some activity of antibacterial activity. We found really poor activity of antibacterial, the MIC. We found also do the same with other species, that's the Witerigia solanacea. We make the phytochemical extraction. screening, sorry. And we have also an extract of the fruits. We found a greater amount of phenolic compounds and total flavonoids and a greater antioxidant capacity compared to the hydroalcoholic extracts of the aerial parts. And at the end, we found that the antioxidant activity is considered low. And we have some of these results. We want the antimicrobial activity and some of the relaxing effect of this muscular contraction. Also, we found anti-inflammatory activity in a PO edema model in rats. And we found that it appears to have a low toxicity according to the OECD protocols. These are social media of INIFAR. So you can follow us. I want to thank you, thanks Veronica, Dr. Pfister, and all the people that help us here. Really thanks. If you have any questions, that will be.
Any questions? Yeah, so
Later.
yeah, later. Okay, so now the next presenter. Carmenza Espadafara, validating the bioactivity of Panama desbaratadora in vitro approaches. I will say some words about Carmen. Carmen has been with us since the beginning, so from 2020, and she is the current coordinator of the Center for Cellular and Molecular Biology of Diseases and senior researcher at INDICASAT in Panama. Her research focuses on topical parasite, drug discovery, communication science, and biophysics of pathogens. Welcome, Carmenza.
¿Cómo? Sí, sí, tiene que estar. First of all, thank you, Veronica, for inviting me yet one more year to this great group of collaborators and scientists. When Veronica asked me to share today, she said, well, everything that you do you have any success in anything cancer? And I said, I work mostly parasites like malaria, leishmania, Chagas, all the ones that Miguel Angel also mentioned, toxo. But cancer, we do less. And then I thought, okay, I think I have two samples that I can talk about. And one of them is desbaratadora. So this plant is really Justicia secundaval, and it has many medicinal properties. So this is it. This is the plant. And the good thing is that it is spread out in many parts of the world, but it doesn't grow that prominently, but in 4 regions: Indonesia, Malaysia, Brazil, tropical Africa, and Central America. So we have a lot of access to this plant. This is the taxonomy of it. This Acanthaceae has it's big, 346 genus, of which one of them, Justicia, has 643 species. So there are 643 Justicias, when you're gonna find a lot of literature on them, but this one, Justicia secundaval, you don't find as much. It's called la hoja de la vida, the leaf of life. Cingamochila must be a Mexican term because I didn't find it anywhere. Cascajera or gravel, desbaratadora, disruptor, and that's, we call it desbaratadora in Panama because they say that it destroys the kidney stones. And I've seen people really get over their pains like in 2 weeks and be almost cured of them. Insulin, because it lowers the glucose levels. Sanguinaria, and it's not because it's bad, it's because the infusion is really dark red, blood root. On cura todo, just the cure-all. If you could see the attributes that have been given to most of the justicias, but this is just for Justicia Val. Menstruation cramps, cold, fever, malaria, whooping cough, anemia, hypotension, kidney stones. It serves against HIV. It has neurological effects. It helps depression. It's a cure-all, really. So, but we didn't find anything cancer. So these are the publications. You see that it has even antinociceptive, so anesthetic properties. These are the solubility of hemoglobin S for sickle cell, hypotensive, antihypertensive, etc. There is another one about something done on diabetic patients with this infusion. So this is what we did. We just got like every time 10 of the leaves. We washed them mildly. We macerated them, and then we let the water boil. When it was boiling, we threw the leaves, and then we let them stay there boiling for 3 minutes, only 3 minutes. And then they were, the infusion was decanted and stored at -20 for experimentation. So it was filtered before being stored with Wattman paper, but then at least once through a 0.2 micrometer acrodisc. And then we had no way of knowing what the molarity or anything is, so we gave the concentration in terms of percentage. So we added 4 microliters to 186, that was 2%, and that's how we made the samples from 2% to 10%. And so these, apologies, because this graph should never have been a line. These should be bars. There's no connection between them. These are just the concentrations, volume of the growth inhibition, or the relative growth that I believe it was 3% of the infusion was causing in MCF7 cells or in Vero cells. And so we saw that at 3 and 5, we got the largest separation of inhibition of growth. So we said, okay, let's stick to these 2 concentrations. I don't know if you see well, but these are 2 MCF cultures. This is the mammal cancer, breast cancer. And this one, it's just been added vehicle. And you see that it's growing. We calculated like 85% confluency. This one has 5% Justicia Val infusion, and it's really disrupted. On the contrary, if you grow Vero cells with that same 5% infusion, they are growing mostly normal. So we wanted to see what happened in terms of death, and we added it and did a caspase activity. So these are just the 2 concentrations, and they look similar. So we'll just explain this, the 3% without the inhibitor, but with the 3% infusion, you find that the growth has been inhibited like to 40%. If you add an inhibitor of caspases, then the inhibition is rescued and it grows almost like 100%. Now, Vero cells, you don't see anything significant happening. So this suggests that caspases are really the main means of producing death in the MCF7 by the application of the Justicia Secunda Val infusion. Now, on the production of reactive oxygen species, which is usually something bad that kills the cells, etc., you see that we used, again, 3% and 5%, and these are the untreated. With 3%, we didn't get most any change in the MCF cell line. With 5%, we just got some significant difference in the Vero cell line, nothing happened. So yes, there is a little bit of production of ROS, but not that much. And then we looked at the potential of the mitochondrial membrane, which usually it goes wrong, it gets depolarized when the cells are going to die. And so I wanted to, looking at this graph, one thing calls my attention, and it's the difference of the potential of the voltage of the membrane of the mitochondria of cancer cells against epithelial cells, Vero cells. So you look at this difference. It's something with the metabolism of the cancer cell, something natural of the cell. I don't get the word happening. In any case, when we treat with 3 or 5%, we get some back to the normal polarization, but not that much. And in Vero cells, well, nothing much happened. This is a positive control used for depolarizing the mitochondrial membrane. So this is just preliminary stories. The thing that I wanted to discuss with you is this is an extract. It's not a compound. It's not even a fraction. It's an extract. There are two ways of going about this when we have like a consortium like this. Do we go the traditional knowledge way? Do we share these with the community or the population that exemplarize this use, medicinal use, and then we share with them through the Nagoya Protocol, everything? Or do we do more examination? We do fractionation. We isolate the compound, we get the structure, and then we try to apply it to a pharma company to see if we can get into preclinicals and clinicals and that. There are two very different ways. If we want to go this way, we need the funding of big NGOs or big organizations like NIH. But maybe there's a combination of both. Maybe we can just do this first and then share some of the revenues with the traditional people. I'm looking forward for the next talk that we are gonna be hearing. So this is part of the data that was gotten of the mass spectrometry on the Justicia Val, the same suspects all over, steroids, anthocyanin, flavonoids, et cetera, et cetera. Then last year, they published this. It was the first time that Justicia Secunda Val was talked about cytotoxic properties. So what they did is they got a line of a cancer of a nasopharyngeal cancer, and they noticed that the infusion, the infusion was lowering the expression of certain enzymes that are the ones that cause the problems in hormone production of the thyroid, et cetera, et cetera. And then they studied also the antiviral and antioxidant, et cetera, et cetera. All of that we have done, Marcelino and I, it's been possible because of the ICBG program, the International Cooperative Biodiversity Groups, that was initiated back in the 2000s, and then it was a program that focused on drug discovery, biodiversity conservation, and sustainable economic growth. And in Panama, it was the Drs. Tom Currais and Phyllis Colley back in 1998. What was that? What did I do? That thought, why don't we do that in Panama? And so they applied to the first time this ICBG was funded. I think maybe Flora is listening to me and she can correct. And then it was funded by the NIH, administered by Fogarty International Center, and Flora Katz was center and key in this. She and a group of 2 other people were leading us, guiding the program. And so we got funding for 5 years, 3 consecutive times. So imagine the amount of funding that that program put into INDICASAT. Most of the first research and even equipment that we got came out of this marvelous ICBG program. That's why I was so happy today when she announced that Panama again is eligible for funding. So in Panama, it was the Smithsonian Institute at the University of Panama and INDICASAT who formed part of this. And while we were in this ICBG program, one of our collaborators from the United States, Kerry MacPhail, found in the banks of the Coiba Island, the one that Marcelino mentioned this morning that is a national reserve, found this algae, it's a cyanobacteria, limbia, where she found a compound that she called coibamida. Look at this structure. I mean, a person, a human would have never thought of a structure like this unless nature gives us the examples, and then we can start maybe working around them. So the coibamide we sent to the National Cancer Institute, and hi, Tanya. And then they made the test on different histological cancers, and they found that the best activity was in breast and in the cerebral central nervous system. And so Kerry MacPhail thought that there is a lot of pipeline compounds for breast, but not for the glioblastoma. And so she decided to focus on studying the coibamida A on glioblastomas, and she did this in vivo test and look at the growth of the tumor in well, maybe almost 18 days, and look at how the tumor was halted, the growth was halted with this coibamide A, impressive. Now, a lot of Chinese groups started looking at the coibamide A, and then they started doing the synthesis, and then they started getting derivatives. And so they have published a lot on coibamide. So I don't know if this is something that we can do. I was thinking, looking at the plant Gaston, that maybe this algae is something that we can grow in a lab, maybe with sea water or something, and then maybe get some coibamide. Even in its natural state, it's good. Anyway, to finish up, so we got some collaborations in this that have been very important through funding from our National Secretariat of Science and Technology. We got partnered with the largest conservation NGO in Panama, ANCON, to study coconut oil and the coconut plant that they grow in a private reserve park that they have in Darien. And through ANCON, then we got partnered with Indigenous Biodiversity Limited, which is a company that it's like a broker for the UK. Through this partnership, we have got the interest of private companies in testing what we get. That this is very important, I think. We first got Oxford Biotrans, a company that does enzymatic the differentiation of the compounds that we give them. This was interested in cosmetics, Coastwall Perfumery. Then we got Biopharm Enterprises. I don't remember what they wanted. Unfortunately, none of these three worked well enough. And then now we have partnered with the University College of London, doing some experimentation in wound healing. And that one is ongoing. But I think that this networking is very important. And finally, I just want to give thanks to the people that work in the Desbaratadora, Sara Rosero, back then, Lorena Coronado, more recently, Doriana Dorta, technician, Michelle NG, technician. This is my lab, not all of them are here. Thank you again, Veronica. Thank you all.
Do we have questions for Carmenza, or you want to do it privately? Yeah. So now we are going to take a coffee break, and we come back in 20 minutes, please. We are back. Now we have with us Dr. Tamara Rubilar from the National Research Council in Argentina, CONICET. And also she is affiliated with the National Patagonian University, San Juan Bosco. She is the director of the Lab of Organic Chemistry and Marine Organism, and also the co-founder of the startup ProMarine Antioxidants. She had been also working with one natural product from sea urchin tested in a clinical trial for long COVID. Here we have her recording.
Good morning to everyone. My name is Tamara Rubilar. I'm a scientist from Patagonia in Argentina, and I will tell you a story about sea urchins and COVID-19 that it was completely unexpected, and for us is still an incredible story. Well, it is important for you to know that I have two of the most incredible mentors, two strong women, Catalina Pastora and Enriqueta Díaz de Vivar, who teach me a lot of things regarding biology and chemistry. And that unique combination of my undergraduate and postgraduate formation helped me to get where we are today. My focus on my study of my research began with echinoderms and focused on physiology, biological chemistry, reproductive cycles, behavior feeding, and distribution. And these wide studies, that research that we did, it helped us a lot on the way. When you need to use a natural source for do some kind of a product, it doesn't matter the end of product, but you need that natural source. It is important first to identify it, to have a continuously and sustainable source, and of course to have a stable quality, which is for itself a great challenge. And when you are talking about molecules coming from the nature and coming from the sea, it is still an even greater challenge. So for those who doesn't know, echinoderms are very well-known animals for food. Sea cucumbers and sea urchin in special are very, very high delicatessen pricey food in the world, especially in the Eastern world. And it is necessary to develop aquaculture for these two types of animals because the overexploitation of the world is enormous. So we were working on this kind of research, and there's a lot of previous knowledge regarding our species and other species, but there are also bottlenecks in the industry. What we did for develop the sea urchin aquaculture was to select a species that it doesn't compete with in a traditional market. We had to develop a lot of technology to finally get an aquaculture, industrial aquaculture. We have to define the markets where we're going to put the products, and we started a new industry. This is like the end of the tail. So you gather where we're going. So we work with Arbacea dufresnii, which is a small sea urchin living in Patagonian seas. And we had all this information that is on the screen before we start thinking about doing some kind of product or some kind of development with this species. What we focus on are special molecules that are called spinochromes, especially echinochrome A, that are found in sea urchins, tests, spines, gonads, and eggs. And these molecules have been studied for over 40 years in vitro, in vivo, and clinical trials. And there are all the drugs in the market in Russia for the heart attack and for glaucoma. So these molecules are already been proved to be safe for human beings. We knew that sea urchins have these molecules, so we did a scientific agreement with an institution from Russia called the Salk Institute, that those are the ones who developed the pharmaceutical components. So we sent our samples, and they were able to describe that we had a spinochrome A and spinochrome E, and echinochrome A in our extracts. So we started a project that is called, what it was called Farmacia, which is the name of the species, where we thought that, okay, maybe we can do some kind of product to be used in the food industry. So we could diminish the amount of antibiotics that the food industry uses for developed food. So that was our main idea when we're starting to do some kind of project coming from the academia to the industry. And we have a really nice original plan where we're going to start a company in 2019, and in 2020, we're going to have a lot of sea urchins, and then we will have to start the proof of concept of our products in 2021 and so on. But everybody knows that COVID hit. And when COVID hit, we all had to iterate and change our focus. So what we did is we iterate our purpose, and we focus on COVID-19, on food safety, because we produce dietary supplements, and to diminish our carbon footprint. So what we did, actually? We basically did aquaculture and do aquaculture. We raise our own sea urchins. We collect the eggs from the sea urchins, and then we do extracts to gather the echinochrome E. In 2020, when the pandemic hit and we did all this iteration, we started to work with a huge project that it was looking for molecules that weren't able to inhibit the SARS-CoV-2. So our hypothesis was, could echinochrome A do this? And we started another project called Grkovic, that is a word of a play of words in Spanish. So we have three hypotheses in the total project, but we are starting with two, thinking that maybe we could inhibit some of the SARS-CoV-2 molecules, proteins, And we did some computational chemistry studies, and we tried to see if we could diminish the viral charge by inhibiting the spike protein and the M-Pro protein. And we did two preprints with this. We presented the hypothesis in the first meeting that we did in this main project that we are all in together. And then we joined to the project that Veronica and Donald invited us to try to send our extras, not only from echinochrome A, but all the ones that we have in our lab to test against SARS-CoV-2. Previously, we did some studies in Argentina, in INTA, working with a bovine coronavirus, and we did two different experiments with a self-pretreatment and without the self-pretreatment. And we were able to see that we were able to inhibit the viral charge in both scenarios. But if we did the pre-treatment, the increase in the inhibition was much, much higher. Afterwards, we sent the samples to 11 Institute, and Dr. Ryan Nett and Dr. Dick Hartman put it in the robots, and they were able to prove that this molecule was able to, again, inhibit or diminish the viral charge in a 20-25%. So this inhibition was consistent and effective in two different viruses of the SARS-CoV and in two different techniques. So by the end of the year, we have a product ready, and we were really, really happy that we were able to try to use this product during the pandemic. However, little did we know that our government and all the governments around the world were focused on vaccines. So after a summer of depression and said, what do we work for? we said, okay, let's try to see what can we do, what can we publish with this information that we have. And we have with another hypothesis that maybe we could help with this syrup that we did with supplements in the cytokine storm, which everybody knows that is huge in the SARS-CoV-2 infection. So our hypothesis said that echinochrome A, which is a highly known antioxidant, was able to increase the mitochondrial function and in such increase the glutathione pathway, inhibit the ROS activity and diminish inflammation. And with this, it could help patients to diminish the effect of the cytokine storm. With this publication in place, Dr. Gabriela de la Raniaga contacted us from the Muniz Hospital because there was a huge problem with COVID sequelae. This is about mid-2021, where all the patients that survived SARS-CoV-2 started to have a lot of complications. And long COVID, we know today what it is, and today long COVID is a known chronic disease, but back then, it is still an unmet medical need, but back then it was starting to get a grasp of what it was. But basically it has around 200 symptoms, but these are the most common ones. And this is a scientific and medical challenge, and the four underlying problems that are behind it are balance. It's need to balance the immune system. There is need to reduce cellular inflammation, to level the oxidative stress, and to activate the functional mitochondria, because now we know the SARS-CoV-2 eliminates the mitochondrial from scratch completely. Almost you get with no mitochondrial. So we said, okay, maybe spinochromes can be a good option to do that because it actually helps with the 4 items. So echinochrome A is able to reduce inflammation, is able to reduce the oxidative stress, and in doing so activates mitochondrial and glutathione pathway. So it's a very good candidate. So we did a third hypothesis, which was, can we diminish the long COVID symptoms? And we partner with 3 amazing doctors from the hospitals in Buenos Aires, Fernando Saldarini, Martin Sibori, and Gabriela de la Raniaga, and with the team with Marisa Avaro, myself, and all the research in our lab, we started this multi-center medical clinical trial. We use a telemedicine platform because we live like 1,500 kilometers from where the clinical trial was placed, and for the doctors was extremely useful. This is a team, it's a huge team of doctors, scientists, and counselors that help the people during the clinical trial because, long COVID is an incapacitated disease and it helps a lot of people to get into depression. So we also take care of the emotional part of the patients. We did a double-blind, multi-center randomized design, which is at the NIH and clinicaltrial.gov if you want to see all the details and the results specifically for this clinical trial. We use this supplement, what is called ECA Marine, where ECA is the short name from echinochrome A. And we tested quality of life, neurological symptoms, respiratory difficulties, emotional state, and biomarkers. And what we found is that ECA Marine was able to recover the patients for most of the common long COVID symptoms. Again, if I'm going to go through the details, it will take me like an hour, but you can see it in the webpage that I just showed. So we were able to develop a product to give a global solution from sea urchin extracts from Patagonia to the world. This is not a small task. It was really, really hard work. It was extremely expensive, and a lot of, a lot of people wanted to try to help during the pandemic. And thankfully, we have a very, very neat product from sea urchin egg extract that today is able to help people with long COVID symptoms, at least at first in our country. This project was a finalist at the Cesar Milstein Awards for Biotechnological Solutions to Human Health. This is part of the team. Behind me is Dr. Richetti. She was the person in charge of recruiting the patients. And then is Karina, I forgot her name. I'm sorry. She was the head of the counselors. And then is Cecilia Carrizo, who was the responsible for the telemedicine platform. So if you have any question, or if you're interested in our project or in our results, please reach out. I'm available. Thank you very much.
Excellent. So now we move to another presentation. This is from Dr. Pamela Leal Rojas and Dr. Addison Altamirano. They are both from the Department of Natural Resources at the Faculty of Agriculture and Environmental Science and Universidad de la Frontera in the south of Chile. Pamela is an associate professor at this department, and her focus is well, she will tell you more about that. Thank you.
Okay. Thank you so much for the presentation and also for the invitation. Okay. I don't know, after the fantastic presentation of Tamara, I don't know what to say. Okay. Can you hear me? So, my name is Pamela Leal Rojas. I am a researcher and academic at the Universidad de la Frontera from Chile. And today I want to show you different point of view about herbal medicine that we are doing from our region, the Araucanía region, and what we can see like an opportunity for human and societal well-being. Next one. So I know all the people say the same thing today, but it's important to say it, right? Chile, our country, is a unique natural laboratory. Also, I want to show you why our region is a biodiversity hotspot. I will show you a few examples of potential of endemic plant for biomedical application. And interesting thing, we have not a lot, 3 examples of the ethnomedicine. So we have in our city a different pharmacy based on herbal medicine. And finally, one opportunity for human and societal well-being. So here you can see our country. We are a very long and thin country. We have a few advantages or very good characteristics. So we have a high level of endemism, a high number of plant species, around 30% of endemic plants. And that is because we have geographical barriers and also climatic condition. You can see at the north, we have the desert, the Atacama Desert, also the Andes Mountain, and at the south, we have the Antarctica, and in between, the Patagonia. So the Araucanía region, we have located in the middle of the south. So for that, Chile is a relevant hotspot for biodiversity conservation and also a source of unique bioresource. So this is our region, Araucanía region. we have lakes, volcano, thermas, and also we have rainforest. It is a hotspot of biodiversity. Here, last year we showed you a video from Rucamanque, but this year we have a book. You can see all the content in this direction. The name of that book is Rucamanque, The Secret Forest of La Frontera, with my colleagues Rodrigo Vargas. Next one. In this book, you can see different pictures about the Condor House. That means the names of the ancestral names. The Rucamanque is an ecological and cultural park. In Mapudungun is Casa del Cóndor. It's an area over 400 hectares. And the important things is a fragment of original forest of the Central Valley in Chile. So they have a unique flora, fauna, and also fungi. But the important thing for our Mapuche people, the ancestral people, is that one. For the Mapuche community, they are living around the Rucamanque, is a very special place because it's or have a spiritual value, because where the Ñens live, that is the soul of the forest and water. The important thing, I want to show you why it's important for us or for our university to have Rucamanque. So for me and for my team, for my faculty, it's a starting point to looking for medicinal plants or compounds. Because there you can find more than 200 species of plants. 21 are tree species, but you can find also 3 small mammals and also fungi. Y frog. So the university is, or for us, is no, the previous one, is a site for conservation and also for research purpose. So a big advance in our country related to herbal medicine is that the Chilean Ministry of Health certified 103 species. So they have, or we have permission to be marketed in the country as a traditional herbal medicine or has a phytotherapeutic preparation. So also we can find all that species in that book. You can review all the species in this site. You can see the infography, that means natural pharmacy. That is a permanent exposition of the museum, the historical museum in Valparaíso. And you can see the science to get through the winter use of medicinal plants. So all the people in our country knows what to use for pain, for fever, etc. Next one. A few examples of plant species and traditional use you can find in this table. So one of them are 3 Quillaja saponarias include. Ogni molinae is a berry, and they are focused on the characterization of the secondary metabolites, and you can see they are found terpenoid, flavonoid, anthocyanins, etc. Next one. So the Boldina is a compound obtained from Peumo. You can find so many papers on PubMed related to use as a herbal medicine. But this one was interesting to me because this group, they are trying to show that using this Peumo, you can see, or you can use for a different use. So like nanoparticles. So it's diversification of the use. Next one. In this table, you can see also matico is another tree, is another source of compound. So many compound are characterized. And you can see that also they are using different extracts and different compound contain also. Next one. A special paper that I will show you is the matico also show activation or induce the platelet by collagen. And you can see the effect and the aggregation of platelet and also the effect on phosphoproteins related to platelet activation. This is a very interesting example because the Quintral, Tristeris tretandrus, is a parasite of, but it has a great value, medicinal and cultural value. And they have in the market, you can find different presentation of them. Next one. But one important thing is my reflection for today is what we are doing or what we do looking for a herbal medicine or compound. Because for our Mapuche people, it's very, very important the relationship between the nature with the energy and also with the healer. You can see Mati. He's Jorge Culaqueo. Is a very important Mati in Temuco. So we can not forget the social cultural context and also to maintain obviously the harmony and productive social and environmental interaction. So Maria Constanza Torri, the next one, please. You can see in this table the most common plants among the community. And 4 of them are very known for us and they are very used for our people. Arrayán, boldo, canelo, chilco, matico, peumo. Next one. So this is an interesting example for us because we have 3 different pharmacy or herbolaria Mapuche, and you can see different contexts, different colors. All the compounds have a different presentation. For example, drops or pills or gels for different disease. Another one or another example is this ancestral Mapuche medicine, Mapulawen, and you can see different also presentation and different use for disease or for cosmetical use. Maquilawen is a Mapuche pharmacy located in the central city of Chile, Santiago, and you can see also different packaging and also performance in the city. Next one. And so in our city also the relationship with the ancestral people is very important. So we have in Temuco the Intercultural Health Maquihue Hospital. And that side, on that hospital, the Mapuche people have a strong participation in the administration and also the health program. It is very close to my university. And finally, how we can contribute to economy or health or support for our women in the region. So we have a project between Universidad de la Frontera y CETSUR, that is a ONG, and the aim of that project was to contribute to the economic autonomy of 21 women from different communities through networking, technical training, and the improvement of the primary marketing packaging. The principal result was biodiversity conservation, promotion of agroecological practice, and also a catalog for the preservation and native medicinal herb. So many thanks for your attention.
Thank you very much, Camila. Do you have any questions? We go to the next. Yeah, Carmen.
I heard that there were some. Thank you, Pamela. I heard that there were some chains of these pharmacies that had not been successful and that had closed, but it's very nice to see that some others have opened. What are your comments on that? I mean, are there some?
They are
drugstores?
growing. They are growing in amount in
Really?
locate, yes. It is around 20, 25 or yes.
Any more questions? Thank you, Pamela. That's it.
Thank you.
So we are getting to the end of the seminar. And for it, we will have some closing remarks. First, we will have Dr. Giselle Tamayo. She's the director of the Center of Research for Natural Products of the University of Costa Rica.
Do you hear me well?
Ya lo estamos viendo, doctora.
Okay.
Bueno.
¿Están viendo bien? Sí. ¿Están viendo la pantalla?
Ya no, acaba de desconectarse.
Ok. Ahora sí.
Ahora sí. Bueno, sí, ahora sí. Perfecto.
Good afternoon to you all. I would like to begin by acknowledging Dr. Veronica Vargas, Dr. Germán Madrigal and his team for organizing this meeting. And secondly, I want to thank the organizers for letting me give some basic information about Ciprona. Ciprona is an anniversary this year, and we hope to celebrate this in the next October. So we are 45 years old. Our mission is to be a center of focus on multidisciplinary work. Of course, our main subject is natural products that are generated by living organisms, their chemical transformations and interactions, biological activity, and potential applications. All for the benefit of the university community, productive sectors, and society in general, aimed at the development of Costa Rica with an international outlook. Ciprona infrastructure is about 14 laboratories. This is, you can see here on the left, up left, that's the main building of Ciprona, where we have these 14 laboratories. We have a computers lab laboratory, and then a meetings room, and then of course the rooms for researchers. And in the bottom, you see the pilot plant that we have located in a different venue. That plant is a polyvalent pilot plant that we have, and we are conducting right now with alliance with the chemistry department. We focus on several issues about natural products. Of course, the main topic that we deal with is the characterization of natural products. Then there is a section that does some organic synthesis of those natural products and their derivatives. There is a very strong laboratory dealing with microbial biotechnology, particularly bacteria and fungi. And there is also a plant tissue culture laboratory that deals particularly with cell culture and in vitro plants. Lastly, we have a small unit doing some antibiotic screening. And with all this, we try to provide innovation into some of our students and some of the entrepreneurs that reach us for different products that they think that they could make. And finally, of course, we provide also services to the small enterprises that deal with natural products. In terms of resources, besides the laboratories that I mentioned before, we have an NMR 600 megahertz. That's very basic. I mean, to have an NMR to do structural elucidation, without it, it's basically impossible to do structural elucidation. So you have to rely on some other collaborators for doing that. We have also a DCMSMS equipment equipped with liquid injection possibility, also with headspace with fibers for doing microextraction. And we have the ion sources we have are, of course, the typical electronic ionization and then the chemical ionization. Last year, we received a fairly very known and very useful equipment, and we are very proud of it. It is the UHPLC Exploris 120 with the Orbitrap technology that allows us to do not only the determination of exact mass of compounds, but also to do metabolomic studies, which is very common nowadays. We have then different laboratories, lab assistants, and then some postdocs that could help us also in the research. These are some takes on the laboratories that we have. Well, we publish, of course, something in the range of 10 to 20 publications per year. These are the publications of last year, and just to give you a brief, a brief example of what we are doing right now. Of course, we collaborate with several institutes within the university and among universities in Costa Rica, and as well as internationals. And we provide services to the industry. And one of the things that I'm very proud of is that we also have a product is produced by the pilot plant, and that actually what we are trying to do is to sell it internally to the university. That is not a commercial product per se, but it's something that we would like to substitute the products that the university purchase in the industry, because they are really good. I mean, that's really good. So that's basically the information of Ciprona that I wanted to share with you. Just 2 minutes to give you just a brief review of whatever we are doing. And if you want to have more information, you can visit the Ciprona website, or you can write to me. I'm really glad to answer, or to Ciprona itself if you have any question about the services that we provide. Now I'm going to give some of the conclusion remarks. As we conclude today's activity, it is clear that natural products continue to play a pivotal role in addressing global health challenges. The presentations we have seen today cover a wide range of relevant topics, from drug discovery for neglected diseases to the industrial production of therapeutic compounds. These talks have demonstrated not only the scientific potential, but also the practical applications of research in this area. Several presentations focus on the application of natural products in disease prevention and treatment. The MEDINA Foundation's work emphasized ongoing efforts to discover and develop new compounds for diseases often overlooked by major pharmaceutical companies. Additionally, the National Cancer Institute's program and novel compounds for cancer prevention and treatment addresses one of the leading global causes of death. The talks on computational biology and high-throughput screening demonstrated how modern technologies are being used to accelerate the discovery of new drugs. These methods offer clear advantages in terms of efficiency and cost, and they are increasingly integral to the field of natural research. One of the key themes today was the importance of the regional biodiversity. In Latin America, Panama, Costa Rica, Peru, Chile, Argentina were highlighted as areas in which untapped resources are potential niches for bioprospecting. The afternoon sessions focus on specific cases of natural products being used to address contemporary health challenges, from cancer research in Panama to COVID-19 therapeutics in Argentina. These examples provide concrete evidence of the impact that natural products can have when applied to pressing global health issues. The industrial application of natural products, as demonstrated by the work of Kilaja Saponaria on QS21, further emphasizes the potential for translated research into commercially viable products. This step is crucial for ensuring that the benefits of research extend beyond the laboratory. I also want to emphasize the critical context in which most of our research operates. It is unfortunate that we missed the conference from Licenciado Jorge Cabrera on the Nagoya Protocol. The Nagoya Protocol on access and benefit sharing is a key part of the conventional and biological diversity and sets the legal framework ensuring equitable sharing of benefits from genetic resources. Nationals' commitments to this protocol is fundamental to maintaining balance between scientific advancement, environmental conservation, and socioeconomic equity. In summary, today's seminars has reinforced the value of natural products research in addressing a wide array of health challenges. The presentations have provided valuable insights into both the scientific underpinnings and the practical implications of this work. I congratulate all the speakers of this session, and I pass this on to Dr. Herman Madrigal. Thanks very much for your attention.
Thank you for your words and the quick presentation. I will now welcome Dr. Herman Madrigal for some last words. He is the director of the Pharmaceutical Research Institute of the University of Costa Rica.
Bueno, sí, está bien. No, está bien. Bueno, muchísimas gracias. Mi inglés no es muy bueno, así que daré las últimas palabras en español. Muchísimas gracias. En especial, bueno, hablo en nombre de la Facultad de Farmacia, del Instituto de Investigaciones Farmacéuticas, del Colegio de Farmacéuticos, del CIPRONA, el Centro de Productos Naturales, de Investigación en Productos Naturales, y de la Universidad de Costa Rica. Para mí es un honor, es una gran felicidad tener a todos ustedes, tanto en la vía virtual o en este lugar, compartiendo. Espero que de esta reunión salgan muchas amistades, muchos conocimientos nuevos, muchos nuevos proyectos y muchas otras iniciativas. El Instituto de Investigaciones Farmacéuticas es un instituto interdisciplinario que busca trabajar en todas las ciencias farmacéuticas, no solo, como en este caso, plantas y productos naturales, sino la cosmética, el desarrollo de tecnología, la bioequivalencia, el control de calidad de medicamentos, la información sobre tecnologías sanitarias, farmacovigilancia y la docencia y la acción social. Tengo que también agradecerle profundamente a todas las compañeras y compañeros que han trabajado por esto. En especial, bueno, a la doctora Veronica Vargas, está presente aquí, a la doctora Jimena Morales, que nos ha ayudado grandemente, a la doctora Daniela Matarrita, Jennifer Sandí, a la doctora Yajaira Quesada, a la doctora Giselle Tamayo y en general a todos los compañeros y compañeras que han colaborado, porque la verdad es que ellos han sido los que han logrado esto. Gracias a nuestros visitantes, bendiciones, les deseo lo mejor, que este sea el primera vez que nos veamos aquí, pero que sea el origen de muchas otras visitas y de muchos otros encuentros aquí o en otros lugares. Nada más queda por decir que ha sido un éxito. Creo que la calidad de los expositores lo ha demostrado y también la calidad de las personas, que es lo más importante. Muchas gracias a todos y que pasen una feliz tarde.
Gracias, Germán, por ser un host de primera clase. Muchísimas gracias, ¿ya? Por la organización, ha sido estupenda. Realmente nos han tratado muy bien. Thank you to everybody who has been following us from Perú, Chile, Ecuador, Colombia, Panama, Costa Rica, too. And from the UK, our friends from the Royal Garden Queue, from the US, and also from Canada. We thank you, all of you, and please keep in touch and send us your thoughts about send us suggestions where we should move forward to. And of course, if you want to collaborate with us, also write to us. It has been a pleasure. Muchas gracias a todos y nos estamos viendo.
Bueno,